A very warm good morning to one and all, honorable dignitaries, distinguished faculties, seniors, and my dear fellow mates. I heartily welcome you all to the day 3 of theme meeting in life sciences. So let us begin with session 8 of today, which comprises of 4 invited talks. It will be my pleasure to invite Professor Prasanna Venkataraman onto the stage to kindly chair this session. And also now I would like to give a brief introduction of Professor Venkataraman ma'am. Dr. Prasanna Venkataraman is the Deputy Director of Cancer Research Institute at Advanced Center for Treatment Research and Education Center, Education in Cancer, Mumbai. She is the convener of Board of Studies Life Sciences HBNI. Also, she did her PhD in 1998 from Indian Institute of Science, Bangalore. Her research interests include protein-protein interaction, typify physical, signaling, and regulatory networks that orchestrate cellular responses. I welcome you, ma'am, and now I hand over the mic to you for further session proceedings.
A very good morning to all of you. It's day 3 and so it's tough to be here at 9:30. So thanks for coming to this session. The first speaker of the session is Dr. Subhash Chandra Bihani. He is from BRC Mumbai. His research interests are understanding catalytic mechanism of enzymes through X-ray crystallography. He uses integrative structural biology to study microbiome proteins. He has been awarded with the Homi Bhabha Award in BRC Training School, OCES. He is of the 48th batch. He has submitted about 21 structures to the PDB. I welcome Dr. Subhash to give the lecture.
Thank you, Dr. Prasanna. First of all, I would like to thank HBNI for coming up with this concept of theme meeting, and also a special thanks to Professor Majumdar and his team for being such a nice host for last few days. We are really enjoying our stay here. I hope we will be invited more to this nice and beautiful place. So the focus of my talk today is on some of the recent structure biology work which we have carried out in our group at RBSST BRC Mumbai. I will not be talking about any particular protein in detail. Rather, I will be giving you a brief overview of different structures which we have done in last few years, so that you get a flavor of what structure biology can do in context of different proteins. At the end, I will also share some of the research work which is not related to structure biology but carried out by my colleagues at RBSST. So at BRC, we are interested in, so as you all know now, by the name ki structure biology is basically understanding three-dimensional structure of protein, and we use X-ray diffraction to determine the structure. So this is a basic workflow for structure biology. So we start with a gene cloned in the suitable expression vector, and we try to express that protein in bacterial expression system. For those protein, we get a suitable amount of pure protein, we try to crystallize it. And this is where the bottleneck in the crystallography is, that very few protein actually crystallizes. So there are very few protein which actually produces in sufficient amount and pure quality, and among them also very few protein crystallizes. So whenever you see a structure in any paper or in a database, you can safely assume that that person might have worked more than 5 to 10 proteins before he may have got a crystal structure. So once we get the crystal, we collect X-ray diffraction data. We can use the home sources, laboratory access sources, or we can come to synchrotron like Indus 2 at RRK. So this X-ray diffraction images is in the form of these different images of this nice looking pattern of black spots, which we call reflections. And then we convert this data into this what we call electron density map. Now this is the experimental result of our experiments, and this map we use as a guide to build a three-dimensional model of our protein structure. And then we represent that model in the form of this beautiful, nice looking pictures which we all see in the publications. So at our group, we are working on many different proteins. Broadly, we can classify them into two categories: proteins which are important for human health, and other proteins which are mostly fundamental basic research interest point of view, and also have some biotechnological importance. So now I'll discuss some of those proteins in a little bit more details. So one of the protein which we have worked actually is HIV protease. HIV protease is a major drug target for developing drugs against AIDS. And this is a poster boy for structure-based drug discovery also. So there are many drugs which have been developed using the structure of this protein, and they are in now clinical use also. But most of these drugs have one particular problem, that development of drug-resistant mutants in this enzyme. So we have determined extracrystalline structures of these mutant proteases in complex with these clinical drugs, and we have established molecular mechanism of this drug resistance. So now we know how a particular mutation results into the loss of affinity for these drugs. So this information is very important if you want to design a new inhibitor. So what we can do, we can either avoid that particular interaction, or we can design our drug in such a way that despite the mutation, those compound remains effective in binding to the active site. We have also taken an alternative approach where we have determined structure of this active enzyme in complex with these two substrates. So it's a protease and substrates are peptide. So we have a series of X-ray experimental structures where this enzyme is trapped in complex with the substrate at different stages of reaction pathway. So using these experimental structures, we have contributed significantly to establish this complete pathway of HIV protease cleavage mechanism. And recently, we have also determined the neutron crystal structure of this enzyme in complex with an inhibitor. Now, neutron structures have particular advantage as compared to X-ray structures, that we can see hydrogen in the neutron structure. So this location of hydrogen we can see in neutron structure, which are rather invisible in X-ray structure. Now, why a proton position of hydrogen is important? Because during the catalysis, the proton transfer takes place in this enzyme. So now, because of this, we have complete understanding of this HIV protease catalysis. Staying with enzymes, we have also worked on many other enzyme systems, and in some of these cases, we have shown that the active site is very different from other known enzymes. And in fact, some of these enzyme systems which we have worked on are the first member of their own family. And they were annotated wrongly in the databases, and based on the structure, now we are correcting those annotations. So first such enzyme class is oxidoreductase. So these disulfide oxidoreductases belongs to thyrodoxine superfamily. You may all be knowing about thyrodoxine. So these enzymes has 2 cysteine in the active site, which remain in the reduced form. So their job is to, whenever they encounter a non-native disulfide bond, which normally takes place during oxidative stress conditions, so they reduce those proteins back to their original state. And there is another enzyme. So thyrodoxine is one such enzyme, and there is another crucial enzyme is glutaradoxine. So these are two important enzymes for antioxidant pathway in cells. So the structure of this enzyme, which we call FRNA, when it shows that there is a thyrodoxine fold, typical thyrodoxine fold with a large alpha helical insertion, and that is an extended C tail. Now this kind of structure with the thyrodoxine fold and insertion was already observed in some other structure, but this C terminal tail was quite unique. And then the story became further interesting. If you see the end of this C terminal tail, there are 2 more cysteines present in the end of the C tail. So we have several different structures at different states, and also we have done extensive mutagenetic studies, biochemical and biophysical studies, and we have shown that these C-terminal cysteines actually interact with this N-terminal catalytic cysteines during the catalytic turnover stage, and they form this intermolecular mixed disulfide bond. Now this kind of mechanism has never been observed before in this class of enzyme. We were the first one to report this kind of mechanism, and also this protein is not alone. What we have seen using bioinformatics analysis, we have seen these homologs of these proteins are widely distributed in prokaryotes. They are not present in eukaryotes, but in prokaryotes they are widely present. But if we look deeper in the distribution, we found an interesting observation. As I said, there are two proteins, thyroidoxin and glutaredoxin, which is essential. But what we found is this FRNE distribution is mutually exclusive to glutaredoxin. So wherever the FRNE was present in a bacteria, what we've seen is that glutaredoxin system was completely absent. Now, physiological relevance of this discovery or observation, whatever you can say, we are yet to identify, but we are working on it further. So maybe, hopefully, in future we will be able to know about this. So another protein, similar story, is an alkaline phosphatase. Alkaline phosphatase are specific phosphatase, and there is no wish to remove terminal phosphate group from organic substrates. Now these are non-specific enzymes. Before we started working on it, there are 8 to 10 structure of alkaline phosphatase was already there, and all of those active site was identical. So in their active site, there are 2 zinc ion and a serine and an arginine, which were employed to bind phosphoryl group. So their active site is pre-configured to identify only the phosphate. So this structure of our enzyme shown that it is somewhat different. So what was the difference here? That it has a combination of 4 residues, an asparagine, an arginine, a threonine, and a lysine, along with 2 zinc ion to bind this phosphate group. Now such combination of these 4 kind of amino acid has never been observed in any enzyme before to bind phosphate group. So this is first such report. And interestingly, when we did the literature survey, what we find that all of these residues were actually present in different member of enzyme superfamily. So like this threonine is present on a different enzyme, that is phosphodiesterase. This lysine is present in sulfatases. Arginine, as I already said, is present in alkaline phosphatase. So SPFP, this active site is kind of a chimeric enzyme. So we did bioinformatics analysis, and we show that again this enzyme is widely distributed in bacterial phyla, absent in eukaryotes. And if we do phylogenetic analysis, the SPAP actually forms a separate group from other alkaline phosphatases. So this is kind of providing us a missing link in the study of evolutionary study of this alkaline phosphatase. So this alkaline phosphatase actually family is actually very much studied to understand enzyme mechanism and how different enzymes evolve. So so this is an interesting area of work, and also this enzyme has some biological and bioremediation potential. So we are working through protein engineering to improve its characteristics so that we can use in those fields. We have also worked on human protein kinase A. So what we did here, we took the crystals and solved in with ATP, and at different time point we froze the crystal in liquid nitrogen and collected data. So we now have a series of results, series of structures where this binding of ATP before the catalysis take place has been observed. So what is happening here is during the binding, what we see, there are subtle rearrangement going on in the active site, especially this serine residue. So this is the first time in these kind of enzymes, this is the first such observation that before binding, the serine residue changes its conformation. So we believe that this information is important if we want to design inhibitor which binds better to the active site. This is an example where we collaborated with other colleagues in molecular biology division, and you will be hearing more about this structure in the next presentation. So this is the manganese catalyze, so which has 2 manganese in the active site. When we determined the structure was very similar to other non-manganese catalyses, but again, structure biology allows you to look deeper into that structure. So if you look closely at the active site, this active site configuration was distinct from other manganese catalyses. Specifically, these 2 residues, a glutamic acid and a tyrosine, was different. And because of these 2 residues, the binding of substrate, which is hydrogen peroxide in this case, is different. So the mechanism of further steps on the catalysis also is likely to be different. So then we studied further. So now we have shown that there are 2 types of active site configuration possible in manganese catalysis, and based on that, all cyanobacterial manganese catalysis can be neatly divided into 2 groups. So one represented by our structure, one is earlier structure. So there are 2 different groups which are evolutionary separated from each other. And also this crystal has provided us with a unique tool. Actually, we kept on hearing one question. People asked us whether you are working on enzymes which are in solid state, in the crystal phase. So they are unlikely to be true because enzymes work in the solution. So this is a very good example to show that if we take this crystal and put in a hydrogen peroxide solution, so it will cleave that hydrogen peroxide into oxygen and water, and you can see this oxygen bubbles coming out of the crystal. So we don't have a movie. I don't have a movie to show you, but this is the picture where this bubble is shown just above the crystal. So it's a very powerful tool to convince people that we are doing something work which is something physiologically relevant. Okay, so this is another structure. So I'm giving you glimpses of different protein structures. I'm not going any detail in any of the structures. So this is a human protein, PSP94. It's a human semen protein, and the structures came out as like primarily a beta sheet structure. And in solution, this protein acts as a monomer and dimer. So at normal higher pH, 7.5 or so, it's a dimer, and when the pH reduces, it becomes monomer. And this is how it forms dimer in the solution structure. So this is H to H beta strand interactions. Now based on the structure, we were able to tell how this switch is taking place. So actually what's happening here, this carboxyl group, end of the carboxyl group at the end of C terminal is forming a hydrogen bond with N-terminal amino group. And in this case, this carboxyl is acting as a acceptor in hydrogen bond. So what's happened at a lower pH? At lower pH, this carboxyl will get protonated. And there are reports and previous studies which shows that in carbonated carboxyl group do not act as an acceptor for hydrogen bond. So that's an observation. So because of that, we believe that this hydrogen bond breaks at lower pH, and now this beta strand cannot stay in a proper orientation. So they will twist, and because of that twisting, the dimer will dissociate and form monomer. And interestingly, this twisted conformation has been observed in NMR structure, which is not our work, it's others' work. And this monomer actually is the physiological relevant important form, and then this form it is now able to bind its physiological partner, and we have a computational model to show how that binding will take place. So recently we have also worked on another human protein. It's a Uvs assay. It's a transcriptional couple repair protein, very essential for all the eukaryotic organisms. So this is a big protein with the N-terminal domain and C-terminal domain interspersed with a large intrinsically disordered region. So we tried to express this protein in E. coli, and despite our huge effort, we didn't get suitable protein. But so we tried different constructs, and when we removed this C-terminal nuclear localized signal, we could purify this protein. So this is the purified protein, and then we characterize the protein, and we show that this Uvs assay binds to RNA with good affinity. We verified using SPR, and this is first such report where anybody has shown that this protein actually binds to the RNA. So now we wanted to map what are the binding sites for this RNA. So we prepared different constructs, and we show that this VHS region binds to RNA, and at C terminal also beyond this DUF domain, this region also binds to RNA. So this is a RNA binding site which are at two different ends. Now there is a AlphaFold model, predicted model of this protein, which shows that in three-dimensional space, these two regions are likely to come together to form a single RNA binding site. We tried a lot to get the crystal of this protein, different constructs, but we failed. At NMR, we tried cryo-EM, but all our efforts are so far not successful, but we are trying further. Okay, so this is one virus now everybody is aware of. This is SARS-CoV-2, which caused COVID. When this pandemic first started in China and Western world, when there was no report in India, we became interested in this virus. Reason of this, because we were previous experience with HIV protease. So this is also an RNA virus. RNA virus produces this protein in the form of polyprotein, and they are immature. So they require a protease to cleave that polyprotein, so the protein becomes mature.
And Subhash, you have 5 more minutes.
How much?
5 more.
5 minutes. Fine. So we worked on this protein. So the problem was that it protein aggregating. Protein was aggregating. So we tried several methods to make it stable. Then okay, this is an example where we didn't get protein crystals. So we have to make mutations, and with one single mutate, we could get the crystal of this protein. So we tried to further screen several inhibitors for it, and finally we are able to find one compound which has good inhibitory potential, and we checked it in animal model also, where it was able to reduce virus load when given oral dose. So these are some other structures. I will not go in detail. So this is our facilities which we are using for characterization. So now I will briefly describe about some other activities which are being carried out in RBSST. So at RBSST, we are very much interested in understanding how radiation interact with biological systems, and also to develop radioprotection and radiotherapeutic compounds. And also we are interested in understanding cancer biology and developing some new treatment methods. So as you all may be knowing that we have a belt in Kerala sea coast which has high background radiation. So we wanted to know how this high background radiation affects human health and the population which is living there. So we have done large-scale epidemiological studies as well as cytogenetics and molecular biology techniques to show that there is no increase in cancer or any other disease, or there is no increase in DNA damages. Rather, what we see is that there is some kind of radio adaptation going on these populations. We also study this radiation effect in laboratory conditions. So we have developed an alpha irradiator in our group, and we also study bystander effect in cancer, bystander effect, molecular mechanism of it, and also how it affects cancer radiotherapy. As I said that we have a large belt in Kerala where the background radiation is high. Reason is that there is a presence of thorium there. So we have a large reserve of thorium, and we would like to use that thorium in our future energy program. So it's very important to know how thorium interacts with the biological system, and if it gets inside, can we remove it? So we worked on these aspects, and now we are successful. We developed a formulation which is more effective in removing thorium even from the tissues. So gamma radiation is most common therapy for cancer, but nowadays this proton therapy is gaining traction. We have a second center in ACTREC. So there is the advantage of proton therapy is that it is more precise. But you posed the question whether this is the only difference or whether protons behave differently as compared to gamma. So we have shown that protons are 2 times cytotoxic than gamma. It also has an inhibitory effect on invasiveness and malignancy of the tumors, and also its from structure is very different. We are also trying to understand the interaction of tumor cells with their immune system. So in this aspect, what we are trying to do is we are trying to find some So in this case, we have identified few molecules which reduces the immunosuppressive characteristics and increases the this one, 2 slides only. Okay. So we have identified few molecules which have immunomodulatory characteristics, and we have tested them in vitro as well as animal models, and we have shown that they are effective in reducing the tumor growth. In order to find more such compounds, we have developed a screening method, and through this screening method, we have identified some novel molecules, and we are testing them in other by other different methods. And finally, I would like to leave you with one example where we have started some 2 decades back using some basic research, and we moved on to clinical stage now. So it's about chlorophyll. It's a chlorophyll derivative where the magnesium is replaced with copper. So we started some 1998. Our first paper came on this compound. So initially we tested in vitro conditions. We found that it has antioxidant effect, it has a radioprotective effect, and it so reduces free radicals. Then we moved on to cell-based assays, animal models, and this and disease model as well. So based on our decades of work, we realized that this molecule has potential to be used. So this molecule has potential where there is a radiation-induced injury in patients. So we went on to do clinical trial with Tata Memorial Hospitals. As the second stage trials are currently. So phase 2 trial is going on in TMH. So phase 2 trial is going on at TMH, and the doctors are happy, very happy with the results. This molecule is able to successfully heal some of the patients which has radiation-induced injury. And also, when this pathophysiology of SARS-CoV-2 virus was clear, we saw then in SARS-2 virus also immune system is involved. So we were convinced that this molecule will also help in the recovery of COVID patients also. So we did some studies, and then finally we also went on the this clinical trial for COVID patient also. So phase 2 trial is already, and initial results are very encouraging. And also not only it helps in the recovery of patients, we also find that this molecule has a direct inhibitory effect on SARS virus. So with IC50 value of 1 micromolar. Further trial, further approval for further trial is now awaited from the government. And these are some of the facilities.
Thank you, sir. Let us thank the speaker. There is time for 2 quick questions. Yeah.
So you said one mutation you have done, and because of that you got a crystal. So can you elaborate, like what could have that amino acid residue has done that it would not go into the crystallization form?
Yeah, this protein has SARS-CoV-2 PL protein has many cysteines. So one of the cysteine is on the surface. So that was the reason that protein was aggregating. So we used DTT in all our assays, but still it was not good enough for crystallization. So when we mutated the serine, protein simply crystallized in 1 day. And also the cysteine is in the active site also. We don't know the reason, but even the active site cysteine when we mutated, that also crystallized. So for that we don't know the reason, but surface cysteine, that's the reason.
And I'll extend onto that. The chlorophyll and SARS inhibition, do you have a proposed mechanism how it could be inhibiting?
Yeah, so this is the work by of my colleagues. So what they have done is that not study molecular biology study, but they have done molecular docking, and they see that this molecule may bind to this main protease of
Okay.
SARS.
So, but your cysteine protease structure is also published.
Yes, cysteine protease structure is also published. So we were hoping that it will crystallize because the structure already there, but in our end it didn't crystallize. Thank you.
Now I request Professor Prasanna ma'am to kindly present a memento to Professor Subhash sir. Thank you, ma'am.
The next speaker is Sri Anand Balal. He is also from BARC. His research interests include molecular biology of bacterial stress responses, genetic engineering, and development of eco-friendly biotechnologies. And he's also interested in transmission electron microscopy to solve structures. He has been awarded with Young Scientist Award for the year 2006 and Department of Atomic Energy Group Achievement Award in 2012, Homi Baba Gold Medal for securing the first rank in BRC training school. He is from the 40th batch. He was a postdoctoral fellow at University of South Dakota, USA. Let me welcome Dr. Balal to give the talk.
Thank you very much for the kind introduction. Good morning, everyone, and thanks for coming here. So at the outset, I would like to thank HBN and the organizers for holding this wonderful meeting, and particularly Dr. Majumdar for having it offline, because we have been used to online meetings for virtually 2.5 years now. So this is indeed a pleasant surprise. And in this regard, I also want to thank Dr. Hema Rajaram for asking me to speak at this conference. So when she asked me, I immediately jumped with joy. It was like getting out of jail card. So with this, I will go on to my today's presentation. I'll be talking on molecular insights into ROS scavenging DNA repair proteins from stress-resistant microbes. But before going into this, I will very briefly tell the different major activities which are going in the molecular biology division at BARC. So the first two activities regarding stress as well as genome maintenance, I'll be talking about in more detail. So here, the two laboratory workhorses are Dinococcus Radiodurans and Anabina PCC 7120. I'll talk about them separately a little bit later. Now, Dr. Sheetal Uppal's group in MBD is working on chromatin architecture, transcription, resolution of RDNA loops, and the bromodomain proteins. Then bioremediation of heavy metals by prokaryotic systems. This work is carried out by Dr. Selina Acharya's group. And Dr. Ajay Saini is working on stress responsive molecular mechanism in plants. Now, before I go on to my main topic, I'll just very briefly talk about these aspects. Okay. Now, cellular and molecular mechanisms underlying uranium bioremediation. So, in this case, different mechanisms such as uranium bioprecipitation, then uranium immobilization, or biosorption by extracellular polysaccharides, or binding, sequestering of uranium by metallothionein proteins in different bacteria such as Dinococcus, Anabina, Serratia, or Chrysobacterium has been used. And not only that, now molecular studies with respect to omics as well as the type 9 secretion system and a detailed study of metal transporting P-type ATPase is also being done by this group here. Okay. Now, with respect to stress responsive molecular mechanisms in plants, as you are all aware, there are multiple mechanisms of gene regulation, which include genetic, epigenetic, alternative splicing, and small and long non-coding RNAs. The present focus of this group, Dr. Ajay Saini's group, is alternative splicing at RNA and protein level. So, AS plays important role in diverse cellular functions and stress responses. What has been seen is that when a stress tolerant variety and a sensitive variety is compared for SODs, alternative splicing is seen to generate higher SOD diversity in stress tolerant varieties. So keeping this in mind, the present emphasis on the lab is to look at the molecular crosstalk between the basic, which is the normal transcriptome, and the alternative transcriptome proteome sets in cellular function and the stress responses. Okay, now I will come to Dinococcus. So here I'll be talking about DNA repair in Dinococcus radiodurans. A brief introduction to Dinococcus. This is an extremophile. So this is the picture taken in our laboratory. So as you can see, it is a tetrad here, and this circle here is nothing but the DNA, which is in form of a toroid. So it forms a nice bangle-like thing. And here you can see the DNA going from one cell to other. Now it is an ancient extremophile, survives extreme dose of gamma radiation. Not only that, it is UV desiccation and nutrient starvation tolerant, and more importantly, it is naturally transformable. So it is easy to do genetics with it. In fact, Guinness book states it as the world's toughest bacterium, because if you see, if at a dose as high as 6 kilograms of gamma radiation, there is virtually no loss in survival. But if you compare them with mere mortals, humans LD50 is around 6 kilogray, so 6 gray. So what we have is a 1,000-fold difference between humans and Dinococcus radiodurans as far as radiation resistance is concerned. Now, what are we trying to study? Okay. So here the questions being addressed in the DNA repair are: what is the role of guanine quadruplex structures in stress biology? Then coming to functional proteomics, the protein-protein interaction in radiation resistance and signal transduction in response to radiation stress. Okay, so I'll just start with guanine quadruplexes. So what are these? So Dinococcus is a GC-rich genome. As you can see there are multiple stretches of G, and intermittent you also have some other residues. So when you talk about the guanine quadruplexes, essentially they are secondary helical structures containing G tetrads. The guanine-rich DNA sequences can fold into 4-stranded non-canonical secondary structure called as the G quadruplexes, which are just shown here. So they are present at telomeres and at regulatory regions, and they form very stable structures. Okay, so the importance of this is that G4 are strongly associated with genomic and epigenetic instability, and particularly when they are not regulated, then things can get very toxic. Okay, now what is the physiological role of G4 DNA in Dinococcus radiodurans? Uh, bioinformatic analysis shows presence of G4 DNA motifs, quite a bit of them. Like if you have the chromosome 1, there are more than 8,500. In chromosome 2, there are 1296, so on and so forth. Now, what is seen is that when you add a particular drug called NMM, which stabilizes G4 quadruplexes, under normal conditions, there is no difference in growth. As what is seen here, in presence or absence of NMM, there is no difference in survival. But the moment gamma radiation comes into the picture, you find a drastic decrease when NMM is added. So this is also reflected here. In presence of NMM during post-irradiation recovery, the growth is reduced. So IR plus NMM, the growth is reduced as compared to, the normal growth, which is definitely better here. So what is seen is that during post-irradiation recovery, NMM sensitizes the bacterial cells to ionizing radiation. Now, G4 DNA are present downstream of several DNA repair genes. For example, the REC-A, REC-Q, SSB, etc. in Dinococcus, all of them contain the G4 DNA motifs in their promoter region. I have just shown the REC-A promoter here. There are 4 guanine quadruplexes. And what is seen is that in response to NMM and IR, the expression of these genes is considerably reduced as compared to the control without NMM. So this indicates that guanine quadruplexes may also have a role in determining expression of a particular gene. Now, coming to the positional effect, so there is an G quadruplex present at -67 of the P REC-Q of the promoter of the REC-Q gene and at -121. So when the stabilizing drugs are added, the G4 stabilizing drugs are added, very clearly you can see there is a decrease in expression here with respect to the control, and in case of mutal, there is an increase in expression. So the position and polarity of G quadruplexes affect the promoter expression. Now, REC-Q helicases and topoisomerase 1B from Dinococcus radiodurans resolve the G4 DNA efficiently. Now, on basis of this, a particular model has been proposed. So Esdsa pathway is the major pathway of repair after gamma radiation. And what is seen is that in normal cases, single strands are generated. They may form G4 motifs. So these are resolved by the proteins what I just talked about. And once these are resolved, annealing and ligation can occur, giving rise to the resistant phenotype. But what happens when NMM is added? When NMM is added, these structures are stabilized. As a result, the polymerase is stalled, and then annealing and ligation is impeded, giving rise to the sensitive phenotype. So the significance of this work is that the G4 DNA regulates gene expression in response to gamma radiation. Okay. Now, the second part is understanding the extreme radiation resistance of Dinococcus radiodurans through protein-protein interactions. So, as I mentioned before, Dinococcus is highly resistant to gamma radiation as well as desiccation. So, what you see, 6 kilograde treated Dinococcus cells, if you inoculate them in medium, they show a small lag, and after that, they start growing almost normally. Same thing what you see here is for desiccation stress. 15-month desiccated Dinococcus culture cells, if you put them in medium, they take off after 6 hours. So, and what is also observed is that in response to both these stresses, there are certain common proteins which are induced. So, for example, you have SSB, SSB1, DDRA, DDRB, and REC-A, which are induced with response to both these stresses. Now, protein complexes from Dinococcus radiodurans cells recovering from 6 kilograde gamma dose were co-aminoprecipitated and identified by LC-MSMS. So essentially, the DRP interactomes were cross-linked in vivo under native conditions. In this case, the 2-hour time point was taken so that the cells are midway to the DNA repair, and all the major gamma radiation inducible DRPs are present. The DNA repair proteins were pulled down by specific antibodies, as is demonstrated here. And finally, an interaction network of DNA repair proteins was generated. So the interaction network displayed interaction between the bait protein and the selected 80 interacting proteins. So the interaction network provides an assessment in one glance. So these 4 particular proteins, you can see the different proteins they are interacting with in one glance. And more importantly, as seen in the Venn diagram here, there are 25 proteins which are common to all 4 interactomes. So essentially 25 proteins interact with DDRA, DDRB, REC-A, and SSB, indicating that all these things are highly interconnected. Yeah. So not only this, the LC-MSMS analysis was further validated by binary interactions using in vitro pull-down assays. And on basis of this information, a model for homology-dependent DNA repair was proposed. I won't go into details of it, but what I want to say is that the model was based on protein interaction data, which was validated in this study and from other studies. So what is seen is that a network of homology-dependent repair pathways function in tandem. In early phase, here you have the SSA, single strand annealing, and the Esdsa pathway. And in the later phase, HR is operational. The homologous recombination pathway is operational. Now, coming to the signal transduction in Dinococcus radiodurans, there is a eukaryotic serine threonine-like kinase which is present. So the question is, in the absence of the canonical SOS response, how is the cell cycle regulated in Dinococcus radiodurans? So what is seen is that there is a novel DNA damage sensor and cell cycle regulatory enzyme called RQKA, which is radioquinine kinase A protein. So what is seen is whenever there is gamma radiation, there is increase in the level of pyrrolopylone quinone, the PQQ, and RQKA has PQQ binding sites. So once PQQ binds to RQQA, the protein is activated. It is essentially it is a kinase. So what it does is it phosphorylates DNA repair protein. So after gamma radiation, you don't want cells to divide. You want division to halt and you want DNA repair to occur. So phosphorylation of these proteins enhances their activity. On the other hand, phosphorylation of cell division proteins and DNA replication proteins, decreases their activity. So it gives enough time for cells to repair their DNA. And once the DNA is repaired, then cell division can proceed. So the significance is alternate DNA damage response regulated by RQKA has been identified in Dinococcus radiodurans. Okay, now I will come to cyanobacteria. So what are cyanobacteria? These are photosynthetic oxygen evolving bacteria. They are most ancient forms of life which evolved more than 3 billion years ago and were the driving force of evolution. So they were responsible for transformation of the ancient reducing world to the oxidizing world. So we exist here because of cyanobacteria essentially in this form. Now, they have colonized virtually all habitats ranging from hot springs, deserts to polar regions. So this is a very interesting picture. This green stuff, what you see in the polar bear, is nothing but cyanobacteria which are growing on the hair of polar bear. And then here you have a rock from a desert in Nevada where you can see it is covered by this green gooey stuff which is nothing but again cyanobacteria. Okay, now how are they grown? Cyanobacteria can be grown in large volumes by using bioreactors outdoors, or in some cases, if you really want huge biomass, you can grow them in raceway ponds. But that is not the way we grow them. We grow them in flask under aerated conditions, or by using a shaker or under stationary conditions, and they can be also grown on plates. So that is what we do. Now this is the organism of our choice, which is Anabina PCC 7120. This is the heterocystis filamentous nitrogen fixing cyanobacterium. So what that means, filamentous means it is present in form of a filament. Heterocystis means you see there is a special cell with two polar bodies at the end. So these are called the heterocysts. So essentially heterocysts are responsible for fixing of nitrogen. And what is seen is that like Dinococcus, these are also radiation and desiccation tolerant. Now as both these stresses also generate a lot of oxidative stress, it is important to study the mechanism which helps in overcoming the oxidative stress in this organism. So the genomic sequence, genetic manipulation techniques are established, so we can do a lot of useful molecular biology work with it. Now here I'll talk about DNA repair first and then the oxidative stress response. So I'll come to the role of the 3 SSB proteins from Anabina. So essentially the database had annotated 2 SSB proteins, SSB1 and SSB2, and if you see compare with the eco SSB, they are fairly truncated. But bioinformatic analysis from our division also indicated the presence of another SSB molecule called as SSB3, which is almost comparable to the standard SSB. So the hypothetical protein, this was annotated as a hypothetical protein. It was experimentally proven to be the full-length SSB of Anabina. Then of the 2 truncated SSBs, SSB1 exhibited NaCl-dependent single-stranded DNA binding, while the SSB2 bound to single-strand DNA only if it was part of a secondary structure. And SSB3 exhibited NaCl-dependent single-stranded DNA binding mode. Now, what is the physiological role of these proteins? To address that, all the 3 SSBs were overexpressed in Anabina. And what was seen was that SSB3, which is shown by pink here, supported DNA repair, while SSB1 inhibited it, the one in blue here. So this indicates that SSB3 is the actual SSB and may play a role in DNA repair.
Dr. Balan, you have another 5 minutes.
Oh, only 5 minutes?
Another 5 minutes.
No, I thought I'm only 15 minutes down the line. Based on the time given here, I'm sorry. We started at 10 o'clock.
20 minutes for each person, so another 5 minutes. Wait.
Okay. So then I'll have to just rush through it. So then this is the SVC and the SVCD of Anabina. Overexpression of these proteins, protected Anabina from gamma radiation. So what is seen is that unlike requirement of formation of SBC-CD complex in bacteria, SBC and SBC of Anabina could function independently and support DNA repair post-exposure to gamma radiation. So based on this, a particular pathway has been proposed where all these different these processes like HR, SSA, ESDSA, and MMEGA are involved in repairing of the DNA. Now I'll come to oxidative stress response. And to this audience, I won't go into the basics because I think all of you are familiar with it. So it is important to note that the superoxide is converted to hydrogen peroxide by superoxide dismutases, and catalases break down H2O2 into H2O, and peroxyredoxins also break down H2O2 into H2O2, but in this case, they require an additional reductant. Now, Anabina has got 2 SODs, several peroxyredoxins, and 2 catalases. The one highlighted in yellow have been characterized in our laboratory. Now, role of SODs were assessed by overexpression in Anabina. So essentially, Anabina's superoxide dismutase was overexpressed along with the GFP. It is important to note that this is only a transcriptional fusion. So you have SOD and GFP mu2 independently expressed as different proteins. So very clearly, both SOD, the MnSOD as well as FSOD were overexpressed. You can see in the thymograms here, in the overexpression strain, you have almost 4 times more SOD activity as compared to the wild type, which is seen here. Same thing for FeSOD. So what was seen was that the MnSOD is located in membrane as well as cytosol. You can see here the MnSOD is only in the cytosol, while FeSOD is present in the cytosol, and MnSOD is in cytosol as well as membrane. So what was seen was that under nitrogen supplemented conditions, overexpression of either SOD conferred tolerance against oxidative stress mediated by methylviologen. Now, hydrophobic region of MnSOD was analyzed in detail. So I'll just come to the conclusion part here. So what was seen is that there is a signal peptide and a linker peptide, and when the signal peptide is deleted, the MnSOD is located only in the cytosol. If you alter the signal linker cleavage site, then MnSOD localization, there is interference with respect to that. And then the 24-27 kDa forms of MnSOD, this is important, they heterodimerize with the cytosolic ion-dependent SOD. So you could have functional hybrid SOD protein, which is made up of MnSOD and FeSOD. Now, what are the evolutionary implications? By this analysis, it was seen was that both membrane and cytosolic forms were present in nitrogen fixers, while only the cytosolic form was retained in non-nitrogen fixers. Now, we'll just briefly talk about peroxyredoxin. So essentially, they have 2 catalytic cysteines which take part in the reaction, and once they react with the substrate, they form either an inter or an intramolecular disulfide bond, and this is resolved by addition of GRX, glutathione, thyrodoxine, or glutaredoxin. So we have worked on the ones which have been highlighted in red. I won't talk about this. I'll just talk about the ALR 4641. Now, this is a 2-cis PRX. As the name suggests, there is a catalytic peroxidatic cysteine and a resolving cysteine. So what we observe is that it is a dimer, but if the wild type protein is a dimer, but if one of this cysteine is knocked out, then it acts as a monomer. And interestingly, all the 3 forms, they appear as higher oligomers on native page, and then they also act as a chaperone. So when you heat MDH in absence of any chaperone, you see the scattering of light, but when any of these 3 proteins are present, the scattering is reduced, which indicates that oligomerization or chaperone activity is independent of the disulfide bond. So essentially, it shows the thyroxine dependent activity, and both the residues are required for this activity. Northern blotting analysis showed this protein to be induced by multiple stresses. Promoter analysis showed the gene to be expressed in heterocysts as well as in the vegetative cells. Okay, so this point is important to remember. Now, 4641 was knocked down using the CRISPR constructs by using, look at this strain here, there is hardly any expression of the protein. Same thing was corroborated by RT-PCR analysis. And what was observed was that if you knock down this 2-cis PRX, the growth is reduced. And not only growth, if you see them under a microscope, the control strands show long filaments, while here the filaments are short. And if you look at the structures here, there you see nice thylakoid structures, while here this is completely damaged. So which indicates that, 4641 is essential to overcome oxidative stress, because in response to H2O2, you see the wild type doing well, but the KD is completely down. So which indicates that this is better. Now, I'll just skip this part. Do I have 5 more minutes, ma'am? because we prepared for 25 to 30 minutes, but maybe I'll just give me 2-3 minutes. So this last part I will just try to elaborate. I'll just try to rush through it with your permission. Yeah. Okay. So I'll just talk about the catalysis from Anabina. So essentially, what we see is that there are two catalyses, but in spite of that, none of them are expressed. So do they have any physiological role? So what is seen was that when you overexpress the catalase in Anabina, it offers protection. So which is seen here. Uh, Now this is an interesting observation. What we saw was that when you treated Anabina culture with salt, and on that if we superimposed the H2O2 stress, then you could see protection here, and this protection was because of expression of the CatB. So this is an enzyme which is not expressed under normal conditions, but when you add salt, this enzyme is expressed, and this, what we see is that this is induced by salt and osmotic stresses, but not by oxidative stresses. Now, another analysis what did was that we identified the promoter, and the promoter activity what we see is absent in the heterocyst. So why is this so? Why is catalase not expressed in heterocyst? You can see the heterocyst here. Here, the GFP fusion shows that there is no expression. So what we believe is that catalase's activity produces O2, which is detrimental for nitrogen fixation. So Anabina chooses not to express catalase at O2 may harm nitrogen fixation. So like I mentioned before, the 2,6-PRX, which is expressed in heterocyst, will detoxify the hydrogen peroxide. Now, as mentioned by Subhash, the CatB protein was crystallized. So I won't talk about this in detail. So what is seen was that the N-terminal region was involved in inter-subunit interaction. So what we did was to analyze this further. We made a series of mutation where this hydrophobic amino acid was mutated to smaller to larger amino acid. And interestingly, what was observed was that only when a hydrophobic amino acid was put at the second position, the enzyme was more compact, as seen by the mobility here on native page, and the enzyme was active. Presence of a smaller residue completely abrogated the CatB activity. So what are the evolutionary implications? So here what we see, amine catalase sequences from database show the presence of only phi, tri, or trip at their second position. The reason for that is clear from our studies, because if these are not present, then the enzyme is very likely to be inactive. Now, just coming to the last part of the talk. So to understand structural meaning into that, what we decided, we also decided to crystallize the CatB F2V. So this was crystallized, and I'll just come to the information. So here, this is the wild type protein, and this is F2V. Very clearly, you see a nice hydrophobic pocket being formed by the wild type protein. But on the other hand, you see a gap here. So the interaction is not so good when you have a mutant F2V protein here. And coming to the activity, the last part. So F2, the F2 is located near the active site of the protein here, Y42. So the reduced interaction between V2 and W43 here perturbs the active site conformation. The Y42 is part of the active site. As a result, the interaction is not proper, and then it perturbs the active site. As a result, it shows reduced activity than CatB. Okay, so with that, I would like to acknowledge our group director, Dr. Himarajaram, and all my colleagues who have, given their input for this presentation, and particularly to Dr. Subhash Bihani for the CatB crystal work. And thank you for giving a little bit extra time. And if chairperson permits, I'll be willing to take few questions.
Thank you, Dr. Balla, but I won't think there'll be time for questions. Thank you for an excellent talk.
Thank you.
I'm sorry, I have to keep to the time.
No problem. I was aware that we'll be given 25 minutes, so I was surprised that 15 minutes.
20 minutes.
You rang the bell.
20.
I request to Professor Prasanna ma'am to kindly present the memento to Professor Anand sir. Thank you, ma'am.
The next speaker is Dr. Tapas Ganjuli. Is that how the name is pronounced? He is from RRCat. Tapas. Dr. Tapas, right? He's there. So this is an acid test for the chair because I have to introduce someone from RRCat with a different field of research. Dr. Tapas Kanjali is the head, Accelerator Physics and Synchrotron Utilization Division. He obtained his BTech and MTech degree from IIT Mumbai in 1994 and 96 respectively. He obtained his PhD in 2006 on his work on pulse laser deposition of, I will spell it out as LNSE. His current research interests include development of semiconductor lasers, growth of 3-nitride semiconductors, and high-resolution XRD studies of epitaxial thin films. I hope I did justice to that. Dr. Tapas, you have 20 minutes. At the end of 15 minutes, I'll get up, so you know there are
20 minutes?
Yes, sir.
Fine, I'll rush through them. I'll skip a few slides in between. Good morning, everybody, and thank you, madam, for an introduction. And I thank the organizers for inviting me here to this team meeting. As a non-biologist, non-life science practitioner, it is a bit of a difficult situation for me to talk about aspects of materials study, which I believe have a lot of relevance to the complete science of studying materials, which includes biology. So let me see if I can make it interesting enough for you to, and try to, instill into you the need for a facility like a synchrotron for several activities in biosciences and life sciences, which you all are involved in. But before I go ahead, it's my present duty to acknowledge. Okay, which one? Sorry, so I would actually start by acknowledging my the complete team here, starting with our director, Dr. Nakhe, then Mr. Tushar Puntambekar, Director of Electron Accelerator Group Board, for their continued support and guidance for this complete activity. All my colleagues in Accelerator Physics and Synchronous Utilization Division for their total dedicated work, which has actually resulted in all this data that I show you here. Our colleagues from BRC Beamline section and few of our colleagues from BRC who are involved in the beamline activities. I have taken some information from them also. And of course, all the colleagues who are involved in Indus operation and maintenance, because without them, all this would not have been possible. My talk, I may very well stress here, is not my personal work, but a collection of the work done by several colleagues at Indus Complex. So this is a flare or an overview of that complete work. I think I have to go back here. Yeah, so briefly the contents are as follows, and I just go ahead to say that X-rays are basically a part of the electromagnetic spectrum. We all know this. But if you look at from the history, X-rays started originally by this discovery, where you can see the image of a hand through opaque material. That is how the people realized that there are X-rays, right? That is how X-rays got discovered by Roentgen. And eventually, today, so this was more than a century back, about 125 years back, and today, X-rays are in a position to go down to imaging to about 100 nanometers. So that's this distance we have traveled in the last 125 years when it comes to X-rays. Similarly, X-rays were used in the early times to detect the structure of materials, and this is the structure of sodium chloride, which is early in the 20th century when Bragg the Bragg father and son duo, when they got this structure determined, they got their Nobel Prizes for just 2 atoms per unit cell kind of a structure. And from there, today, we have the structure of ribosomes, which have, well, more than about 5,000 to 7,000 atoms. So you have huge structures which are being solved today in comparison to what it was 100, 120 years back, which were really very small. And why was all this so? All this is simply because of the fact that the secret, in my opinion, and as most people would agree with me, lies in the progress of X-ray techniques and the quality of X-ray sources. Extremely important that the quality of X-ray sources have improved millions of times from what it was 125 years back, and that is what is the basic motivation of this present talk today. So, when X-rays fall on matter, they interact in numerous ways. These interactions primarily are interactions of X-rays with the electrons in the material, and you could have several processes going on simultaneously. You could have fluorescence, which is again emission of a X-ray from the material after being excited by an external X-ray source, which is a characteristic emission from the particular material. So you can do composition analysis, composition mapping. You can have absorption of X-rays, which gives you a very good idea about the kind of material that the system is composed of, both in terms of its composition as well as in terms of its chemical state, also in terms of the structure. Similarly, we could do diffraction, which I showed you an example just now, which essentially gives you the arrangement of the atoms in space. You could also do imaging, which can give you the way the system is organized at the nanoscale. We have techniques of scattering called small angle scattering and photoemission spectroscopy. I'll not talk about these. I'll only concentrate on these three techniques today, which generally cover the complete spectrum or the major spectrum of its applications. So, when we look at X-rays, we want a good X-ray source. I said that over the period of last 100 years, 150 years, what has improved is basically the quality of X-rays. Am I audible? I think it's fine. So what has improved is the quality of X-rays. So what do you mean by a good X-ray source? This is very important. First, we should know what a good source is, and then we decide on which source to go to, right? So the photons must be, large number of photons must be coming out. They should come out from a small area. They should have a small angular cone, and they should be directional. So if we put all these together into one figure of merit, we call it brilliance, which is photons coming out per second, per unit area, per unit angular cone in a small bandwidth, energy bandwidth. So this is called brilliance. So we need this quantity to be as high as possible to help you to do good and meaningful experiments. So you have a low brilliance source like a bulb, we all know this, in the visible light I'm talking about. We have a very high brilliance source like the lasers, we all know about it, in the visible light, in the visible spectrum again. And this essentially shows that if you have a large area and large divergence, you may have a lot of photons, it will not do a good job for you, whereas this one definitely, as you know, does a lot of job for us, and a lot of spectroscopy today depends on lasers itself. So high brilliance is an extremely important characteristic that ensures that all the emitted photons can be focused on the sample. I should not say all in principle, as much as possible, is actually focused onto the sample for your experiments. So this is a very important aspect. You need a brilliant source. Fine. How do you get the sources? Well, in the laboratory, you have an X-ray tube. You all are aware of this diagram. This is the class 12 textbook diagram. But what is the typical size of this source? Well, if this is about 6 feet tall, a high instrument in the lab, then this is just about 1 feet or so. So that is the size of a lab X-ray source, which is very common. Many people use it. Many of you all may have used it also. But this is a source which does not have much brilliance as compared to what we call a synchrotron. So what is a synchrotron? That's what is our next question. Well, if you have charged particles which are accelerated, then they emit radiation. This is a fundamental theory of physics. We don't question it. This comes from the basic Maxwell's equations. We take it as an established fact. So if you have charged particles which are accelerated, they emit electrons. Then what is synchrotron radiation? Is essentially the radiation emitted when relativistic particles, that is particles which are moving at a very high velocity, very close to that of light. If then such charged particles emit, are bent by a magnetic field, which is again, when bending of a particle would mean acceleration, and that acceleration generates radiation, which we call as synchrotron radiation. That is what is called synchrotron radiation, and synchrotron radiation is the heart of what the talk here today is. So how do you do it? You actually inject some set of electrons into a ring, and you have these magnets around here. This blue color are the magnets which are bending the radiation. This could be a different kind of a magnet structure like this, which is an alternate periodic magnetic structure, or you could have magnets here, which are basically just a North Pole, South Pole configuration of magnets. So when the electron beam bends, let us say it is bending here in this magnet, it is emitting radiation, and this is what is called the synchrotron radiation. Similarly, if you have an alternating magnetic field up and down alternating magnetic field, then the electron would just wiggle through and give you a radiation, which is again because of the acceleration of the particle when it's moving through this magnetic field. This is called an undulator, or we can briefly call them as insertion devices. They also give a lot of copious sources of X-ray radiation or synchrotron radiation, which is also used for several of our experiments. And important to notice that if you look at the spectrum, the spectrum is typically sharp and this spectrum is typically very broad. But these are the characteristics of a radiation. What is important is that all these bending magnets and these insertion devices emit simultaneously, and any modern synchrotron, you could have as many as 50 simultaneous sources of X-rays. So that is what is a synchrotron about. And how's the size of the synchrotron? Well, if you look at a synchrotron like the one we have in RRCat, it's an electron that rotates in a ring which is 173 meters in circumference. That's much, much bigger than what you see in a lab, obviously so. And you look inside, well, you have lots of equipments, these bending magnets and all these things. The electron is essentially moving in that ring, which I showed you in the previous diagram, is basically physically shown in this diagram here. And these are really big size equipments. I mean, you can see a small computer in front of it to give you an idea of how big the systems are. This is only a very small part of the ring, maybe 1/10 or 1/15 of the ring. And if you look at a state-of-the-art synchrotron today abroad in France, ESRF, it is about 900 meters in circumference. These are really big equipments as compared to a laboratory source. And why do we need such big equipments? Well, we'll come immediately in the next slide, where we can see the brightness or the brilliance. They are equivalent words to use. If you look at the brilliance, you will find those of the lab source are only of the order of 10 to the power 6 in the standard units, whereas a synchrotron source would take you anything around 10 to the power 14 to 10 to the power 18. And even today, with the most modern synchrotron sources, this number has been pushed up to 10 to the power 21 or even 10 to the power 22 photons per second per millimeter square per millirad square in 0.1% of the bandwidth. That's really huge number of photons as compared to what you can get in the lab. So that is exactly what brings us to the need of a synchrotron to do several types of experiments which you cannot do in a laboratory. So I hope I've been able to convince you that yes, there is a very powerful source of X-rays which you all can use. And let us go ahead and see what do we have in our center. In our center, we have an Indus accelerator complex. Some of you who would be interested could also make a visit tomorrow to this place, and I'll be more than happy to take you around and show you the facilities out there. So this is a electron storage. This is the ring, which is what called the Indus 2 ring, where you have an electron of 2.5 GeV at a maximum current of 200 milliamps, which is rotating inside it, moving, bending at these bending magnets, giving radiation, X-ray radiation, which is being used for experiments. As I told you, that these experiments are carried out parallelly in all these experimental stations. And as for today, so let me tell you, very important, that this is a national facility, a facility available to researchers from all over the country, to everybody here who have come from other centers other than our RRCat, and also to anybody from all over the country, including industries, by the way. Right. And today, typically, we have around 950 user experiments carried out in a year. We expect to cross 1,000 maybe by the end of this year. And we have academia and industry both coming up and using these facilities, especially the pharmaceutical industries, the primary industry is coming today to use these facilities for their product developments. Okay, and if you look at the brightness which is available from the Indus, so the brilliance which is available from the various Indus, various parts, various sources within Indus, like the bending magnet, you can see that the range of photons could go anything around 100 eV to about about 30 to 40 kilo electron volts. And these insertion devices, which are shown here, have obviously much higher fluxes, much higher brilliances of the order of 10 to the power 17 in the standard units of brilliance. So this is what is available to you at Indus. Well, this graph may not make much meaning because you don't use the X-rays like this. You use it in a slightly different way. How do you use it? Well, you do it through you have to actually get a single wavelength and focus your X-rays. So you use monochromators, you use mirrors, just like you use them in the laboratory for visible light, and you use them to essentially take the X-rays from the source to your experimental station. And what you do here is what is called a beamline. This is basically a schematic of this complete arrangement, which is taking the X-rays from the source to the experimental station. And this is how it looks inside. Okay, So what are the available facilities to you all to do experiments? You can do a large number of experiments in Indus. And today I'm going to talk about very briefly about imaging, scanning X-ray absorption spectroscopy, X-ray diffraction, and just touch upon protein crystallography. As a non-biologist, I will not be able to talk much about it, and you've heard a lot more of it in yesterday or in the evening lectures, and today also you heard a lot about the proteins and their structures and the relevance of their structures. They can also be obviously determined through synchrotrons. Okay, so the X-ray diffraction, well, most of you know this, it's textbook stuff, so I will not go into this, but I'd like to tell you that X-ray diffraction is a beautiful footprint of a material's structure. That is, if you have carbon arranged in the form of a diamond lattice or in the form of a graphite lattice. Both of them are carbon if you analyze the chemistry or the chemical nature of it. But if you look at the diffraction data, you would find that one of them is very different from the other. So the diffraction data is a fingerprint of the crystal structure, although the chemical structure could be the same. And same example I show here for cobalt, which could crystallize into a cubic system or a hexagonal system, and you can see how different the X-ray diffraction patterns are. So that is why X-ray diffraction is a footprint of the structure of the material that you're talking about. And well, these are the facilities available to you, BL12, BL11, and engineering applications beamline BL2, where you can do lots of powder diffraction measurements. And let me give you an example, a boring example from a biologist's point of view, but important because I'll tell you why. See, you have materials like bismuth ferrite, which show very different physical properties. Let us just say this is a polarization characteristic. Let's go much into details of it. They are different, but why? They are all the same material. They all have bismuth, iron, oxygen in the same ratio. Why are they different? If you do a laboratory measurement, which is shown in the black line here, you don't see much difference between this and this. But when you do a synchrotron data, you can see the synchrotron patterns are actually quite different with a large number of small peaks, which are basically what we call as minor phases. These minor phases decide how the physical properties are different. And now why is it important for you? It's important for everybody here, and we also realized this when we started interacting with the pharmaceutical industry, that they are interested in their APIs and a very specific phase in the API, very specific polytype in the API, which is only going to have the desired characteristics of desired behavior in the human body. And that is why they're interested in seeing that these minor phases are absent. They could not see it in the laboratory source. They need to come here in a synchrotron. And that is one of the ways in which we have interacted extensively with this sector. I think maybe this could be of some interest to anybody here. This is the basics of protein crystallography. I'll not talk too much into details except for saying that we can solve protein structures, very complex structures, just like the ones I showed initially, the ribosome structure also. So all these kind of structures can be solved here and are being solved here routinely at our protein crystallography facilities. Then I talked about X-ray absorption spectroscopy. Not too much into detail, but just to say that the absorption features that you see, absorption is a function of the incident energy. These features that you see is a function of the arrangement of the atoms about a central atom. Extremely important. These arrangement of the atoms about a central atom decide what are these features in the absorption spectra. For example, if you look at different materials, which are all zinc oxide with slightly different doping and zinc metal, you can see the absorption spectra are quite different. So this is a very important way to identify the environment of a material or an atom in a matrix. And again, this I can skip this actually because of interest of time, but I'll just show you the lots of facilities available to do X-ray absorption data, either in the hard X-ray, where you can talk about the absorption from the K edge of metals primarily, and you can also do in the soft X-ray spectrum, part of the spectrum, where you can see the K edge absorption from materials like carbon, oxygen, nitrogen also. So you can do all these measurements, and people are doing it regularly out here. So one example I want to show you, this is from a paper which came out in Dalton Transactions a few years back from our colleagues in BRC, who are interested in finding out how this complex structure essentially traps the actinides, which is a useful activity for our radiation programs of our department. So what could they could actually see this structure, they could actually understand the mechanism of how this structure traps the heavy actinide in the cage. So the examples of fluorescence would also I would like to give you, but then very briefly, So fluorescence, as I am telling you, is a technique which is used to detect the elemental composition and down to fractions of a million, parts of per million. That's the sensitivities. And this is an example where we show what are the different chemical compositions of a normal water, groundwater and coconut water, which is, you can see, these are all milligrams per liter, and quantities, as small as fractions of a milligram per liter could be very easily determined with high degrees of accuracy. And this is another example where gallstone compositions were also determined. Okay, I show you an example of the Chandrayaan mission. Maybe this is not going to be of much interest, but to tell you that synchrotrons have got a lot of relevance to do things which are other than what we do in the mainstream material sciences. If anybody is interested, I can talk about this in detail subsequently. And finally, I would like to show you some examples of X-ray imaging. As I started with the first slide, I showed you that X-rays got discovered through an experiment of imaging. This has gone a long, long way, and today we can image down to fractions of a micron to nanometers. And these are some of the images shown in our imaging beamline. This is the image of a kidney stone. This is the 3D image of a silicate foam. All these overall dimensions are just about a millimeter. And these are some other examples of the effect of some medication on the bone structure in a zebrafish. This is an example for bone scaffold material. And the porous structure in that, you are actually looking at pores which are fractions of a millimeter, about hundreds of microns in sizes. You can talk about microstructure and porosity analysis of glassy carbon. And this is another example of a copper sulfate porous 3D printed materials for bone tissue engineering. So various kinds of examples exist which are related to imaging of the materials through X-rays. And so to summarize, well, these are India's only two operational synchrotron radiation sources today are Indus 1 and Indus 2. These are national facilities, and we welcome users from all over the country to come here. As I said, around 950 users are typically using these beamlines annually, which we expect, of course, to increase to more than 1,000. Of course, this is the utility is cross-disciplinary, no boundaries of disciplines. Everybody from physicists to biologists to archaeologists to chemists, everybody has found this to be very useful. Total number of 17 operational beamlines on Indus 2, 7 on Indus 1. I didn't mention much about Indus 1 today due to the lack of time, but when you are visiting tomorrow, maybe we can give you a better flair of that also. And for more details, kindly visit the website of our center. This is all the details of the beamlines and the facilities. And with this, I would like to thank you all. And thank you, ma'am. I hope I'm on time.
So I did give you 3 or 4 minutes extra.
And I thank you for that.
And
in the interest of Indus, I thank you for that.
So anyone has any questions? We can take 2 quick questions.
Yeah, we have a question.
Sir, good talk. Sir, I want to know that your X-ray imaging, that you analyze the nutrient composition of water and coconut water.
That was fluorescence.
Fluorescence, yeah. So it's a non-destructive way?
Absolutely non-destructive.
Thank you.
Absolutely. You just have to take the water, I mean, the liquid sample is essentially put on a glass slide or on a flat place. It is just solidified. I mean, this liquid is made to evaporate off so that all the chemicals are actually settling in there, and then you take a data. So that's about it. So that way it is non-destructive. Excuse me?
If I put some seeds or some grains,
yes, we can do that. Depending upon what is the matrix and what is the material, we can either take the seed as it is, or we may have to, make a piece of it or something. But it all depends on the material, the matrix, the compositional levels you're talking about. If it is just about, let us say, fraction of a percent, then you can just do it without any preparation. But if you're going down to parts per million, which is obviously the strength that we want to exploit here, then you may have to take a piece and then we kind of make it into a solution, and then we dry that solution on a flat substrate and do what is called a total reflection XRF. So, I mean, with your interest, we can discuss these points and solutions.
Last question.
So very nice talk. So I have just one question. So are there any workshops that are like organized by RCAT to familiarize people or students?
Yeah,
who are
we did
not
have
ex-
we had lots of focused workshops, maybe, you know, until about 2015, 16, when we had, you know, initially started this user program. We had lots of focused workshops, including diffraction, crystallography, fluorescence, and its absorption and all that. In the recent past, we've not been having too many workshops. 2 years were down with COVID and all that. Well, if there's a need, we can plan out something like that, surely. And we are open to all discussions. So all the contacts are there on the website. I mean, I'm here. We can talk after this also. There's no problems. And so specific problems we can address. Yes, I agree that we should have more workshops. But then, yes, we did have a lot of them in the till about 2015, 16. We had lots of them. Then we kind of tapered off, and well, we were involved more as the user numbers increased. We kind of decreased this number of workshops. And last 2.5 years, practically nothing because of COVID. And then, well, now this is one of the ways to start again.
Thank you.
Thank you very much, Dr. Tapas, for an excellent description of the facilities and the potential of that. Thank you.
Thank you very much.
Now I request Professor Prasanna ma'am to kindly present a memento to Professor Tapas sir. Thank you, ma'am. Thank you, sir.
The next speaker is Professor Vadan Nagar from BRC. Before I call Professor Vadan to give the lecture, my sincere apologies. I messed up the order because there are two different notes that are there with me. Dr. Vadan's research interests include quorum sensing and biofilm in foodborne pathogens, biocontrol of foodborne pathogens using bacteriophages, radiation processing of food. His scientific recognitions are as follows. He has a Korea Atomic Energy Postdoctoral Fellowship at Kere, South Korea, and an international travel grant by DST to attend ISAP France in 2014. He is also a CSIR JRF Fellowship holder during his PhD. Dr. Vadan? And you'll have 20 minutes. At the end of 15 minutes, I'll get up.
Good morning, everyone. First of all, I would like to thank the organizers for giving me an opportunity to present our divisional work in this team meeting. So I will start with an overview of various R&D activities going on in Food Technology Division at BRC. As we know that based on the FAO estimate in around 2018-19, around 30% of the food grains and 40% of the horticulture produce in India is either lost or wasted during post-harvest processing, and we need a mechanism or the technology by which these losses can be reduced. So we can use a irradiation method by which we can reduce this post-harvest losses. So food irradiation is a process in which the food products are exposed to ionizing radiation in a controlled manner. And depending on the type of food and the objective of our food irradiation, we can optimize the dose for that. And one of the major thing is that the food never comes in contact with the radiation source. This technology provides various advantages, of which one is it is highly effective and it is non-residue forming technology. It is safe to workers and environment. It can be applied to pre-packed commodities. It can penetrate deeper into the food commodities, and it is a cold process mechanism technology in which it does not affect the organoleptic properties of the food material. So overall, our mandate of our division is to use the gamma radiation for food security, trade promotion, and food safety. This food irradiation can be done by radioisotopes, in which cobalt-60 is the major one, and in the machine-based, we have electron beam as the radiation source. So this is the schematic diagram of the gamma radiation facility, in which the cobalt-60 source is immersed in 6-meter deep water. And when we load the pre-packed food products in this source is shielded by a thick concrete wall. And when the food products come inside this shielded area, then the source comes up and it irradiates the food product. And after a certain dose, the dose which we want to give, it is depending, dose is equal to dose rate into time, and depending on the dose which we want to give, we optimize the time. And then after irradiation, the source goes back to the water and the food products are unloaded through the conveyor belt. The wholesomeness, nutritional adequacy, and safety of irradiated food has been extensively studied, and this technology has been approved by various regulatory bodies like WHO, FAO, IA, and FSSI. And many countries, around 60 countries, including India, have approved radiation processing of over 100 different food items. Recently, class-based permission for the irradiation of different food commodities have been approved by Government of India and FSSI, and a gazette notification has been released in year 2016. And based on that, all the different food products have been classified into different classes, and depending on for the purpose, they have been given the minimum and maximum dose. So radiation processing a technique which can be used for different purposes, like sprout inhibition of onion, potato, insect disinfestation, shelf life extension, hygienization, and for the quarantine of fruits. So now I will be presenting the various food products and the technology developed by FTD in the last 5 to 7 years. So one of them is the shelf life extension of lychee. As we all know that lychee is a very perishable fruit, and it is mainly grown in the Bihar region. So, but it has certain limitation. It gets spoiled very fast, and the pericarp of the fruit gets in brown in color very early. So we have developed a drip treatment technology by which we can retain the nutritional quality of the fruit along with the shelf life extension 45 days, and a lychee treatment plant has been established in the Muzaffarpur, Bihar. As we all know that the spices are often contaminated with various insect eggs and the various pathogens which spoils them. So we have developed a radiation based technology which can increase the shelf life of these spices by more than 1 year without affecting its nutritional qualities. And similar way, this has been here I have shown the turmeric and black pepper, but other spices like cumin, coriander, red chili, and red ginger can also be irradiated and preserved for a longer period of time. We can also see a lot of insect infestation in cereal and pulses, and it spoils our food product, and it is causing a lot of damage in the Food Corporation of India. So BRC has developed a technology using gamma rays which can extend the shelf life of this cereal and these cereals and pulses by around 6 months to 1 year. Under IAE project, there was a need to develop food products for the military astronauts and the various calamity affected people. So BRC has developed different food products, and among this, this ready to eat stuffed bag food product was developed, under which DA has supplied this stuffed bag food to flood and landslide affected people during floods in Himachal Pradesh in 2018, and various other shelf stable food products like puran poli, pulao, paratha, and this instant fish soup have been developed, which can be stored for a longer period of time. As we all know that fruits are very rich in nutrients, and they are quite perishable. So BRC has developed preservative-free technology, radiation-based technology, by which we can preserve the jamun, strawberry, and other fruits for around 6 months without affecting its nutritional and texture qualities. According to our Ayurveda, this Karela, bitter gourd, and the fenugreek, methigreek seed have a lot of medicinal values. However, this type of products are not accepted by the consumer due to its bitter taste. So BRC has developed a technology for the production of debittered gourd juice and the fenugreek seed extract. And similarly, we have also developed a technology for the enzymatic extraction of banana juice from banana. And all these technologies can be used in different food industries. So overall, in the last 5-7 years, BRC has FTD has transferred around 21 technologies to different private parties and rural entrepreneurs for its commercialization. And at present, we have 25 food irradiation plants across India in different parts of the country, and many more such plants are going to come in the near future. Here we can see the cobalt-based food irradiation plants across India. Here we can see the earlier they were mainly concentrated in the western part of the country, but nowadays we can see that different states are also taking up this irradiation-based plants. So in India, in US, there is a lot of demand for the Indian mangoes. However, US doesn't accept the mangoes which are not irradiated due to the presence of seed weevil in it. So FTD has developed a technology by which we can overcome the quarantine barrier which is there for the mangoes. And using this technology, we have started export of the Alfonso mangoes since 2007, and the quantity of the mango which are exported has significantly increased. However, this export was mainly using air cargo. However, there was from the farmers and from the various traders which were involved in this trade, they wanted an alternative in which if they can use the sea route, then the cost of transport can be reduced. So FTD has developed a technology in the last 3 to 4 years in which we have used some method by which we have developed a technology by which the shelf life extension of the mangoes takes place by around 25 to 30 days, and during this time these mangoes can be exported to USA by sea route. So it will reduce the cargo charges by around 1/8 of the charges which are there for the air freight charges. And the successful demonstration of this assignment or the export of this technology was shown in June 2022, and this consignment was cleared by US regulatory agencies, and this technology has opened multiple avenues to the stakeholders for which are involved in the mango trade, and it is expected to boost the export quantum. Under Operation Green, Government of India is laying lot of importance on the preservation and shelf life extension of 3 vegetables: potato, onion, and tomato. So FTD has developed a technology for the radiation preservation of vegetables for a sustainable domestic supply, in which a very low dose is given to this vegetable, and along with the optimum storage condition under a low temperature and ambient humidity, they can be stored. This potato and onion can be stored for around 8 months, whereas the shelf life of the tomato can increase by 60 days. Then our next objective was to show the from lab to actual conditions. So we performed the commercial trial for the preservation of potato, and in this around 28 tons of potato were irradiated at our MSMB facility, and they were stored at low temperature. And after 8 months, we can see that non-irradiated potatoes show the sprouting. This sprouting was observed in around 2 to 3 months, whereas in case of irradiated potato, there was no sprouting, and they were in good condition even after 8 months. The large-scale irradiation of onion was also done at our Krushak Lasselgaon plant, in which the onions were irradiated and they were stored in a low temperature along with relative humidity of around 65 to 68%, and it increased the shelf life of this irradiated onion to around 8 months. One of our group is working on the development of functional foods and assessment of its health benefit. Functional foods are the foods which provide additional health benefits in addition to its nutritive value. So we know that gluten, which is present in wheat, is highly required for the making chapati and bread. And but in few people it causes celiac disease. So we wanted to develop this gluten-free multigrain premix, by which the people who are sensitive to this gluten, they can consume it. So we have prepared this high fiber gluten-free multigrain premix by using the radiation processed dietary fiber. And when we did the proximate composition analysis, we found that the protein content of this premix was similar to the wheat flour. And on looking at the brittleness texture, we found that there was a reduction in the brittleness of this fiber fortified dough, and it was improving the rolling ability of the dough, and even the sensory analysis of the prepared chapati was also in comparison or similar to the normal wheat chapati. And based on all these things, the technology has been transferred to various parties. India is the diabetic capital of the world, and mainly we use wheat in that. So we wanted to develop low glycemic index wheat chapati, and here also we added radiation processed dietary fiber so that it will reduce the glycemic index of the wheat. So here the dietary fiber is irradiated, which reduces the gel strength of the fiber. So overall, it will enable us to add more quantity of fiber to the normal wheat flour. And on doing the sensory quality, we found that its sensory quality improved, and the clinical studies with human volunteers have shown the successful development of low glycemic index chapati. As we know that inulin is a well-known dietary fiber, and we wanted to study how the radiation processing will affect its properties. So we found that the irradiated inulin, the solubility increased, whereas the viscosity and the molecular weight reduces. We also checked the changes in the biological functionality, and we found that there was increase in vitro antioxidant activity, and it also allowed the effective growth of various gut microflora. So this irradiated inulin was used for the development of various functional foods. So here we have developed a Synbiotic Jelly. Synbiotic means the presence of both the prebiotic and probiotic in the same food. So we have developed the Synbiotic Jelly, which was showing the same organoleptic analysis, whereas we have also developed the Synbiotic Sweet Yogurt, and here also it was of the similar quality. And here we also observed that the prebiotic culture was surviving in these conditions. Many of the fibers, dietary fibers and proteins which we consume, they are not digested by the gut enzymes, and these are available for the gut microflora to grow. So we wanted to check we wanted to compare the growth of this various gut microflora into the normal dietary fibers and the irradiated fibers, and we can see that this gut bacteria were able to grow quite well in the presence of irradiated fibers. To further study how these bacteroids, which are the gut microflora, they can use this dietary fibers and proteins. So we have isolated the OMVs from them, and we did the proteomic analysis, which showed the presence of uncharacterized endoinulinase in presence of inulin, and metalloprotease in case of when they were grown in gluten, and aminopeptides when they were grown in soya protein.
You have 2 minutes.
Biopolymers are regularly used in the food industry. So we have basically methylated these biopolymers, and this methylated guar gum had various benefits, and this methylated guar gum was used for making film along with the nanoclay, and they were also successfully used for the flavor encapsulation. Similarly, fish oil was extracted from the fish waste, and here also we found that in the alginate beads, they were retaining their functional properties during storage at room temperature. We have also developed a technology for making thermoplastic polymer composite, in which we have used cellulose from the waste of this sugarcane industry, and we have combined it with this PCl, and here gamma radiation was used for the cross-linking, and we found that this polymer had a higher strength, and this cellulose-based biodegradable polymer composite can be used for food packaging and various biological applications. In our FTD, this xanthomonas is used for the production of xanthan gum. When we grew it in the rich media, we observed that it reached a certain cell numbers, and after reaching the cell number, it suddenly started dying, and later on it was observed that it was producing some caspase-like molecules, and it was also showing the hallmarks of PCD. And later on, since it was related to the cell number, we wanted to know whether it is related to the quorum sensing. So we have identified 3 different molecules which may be having a role in the induction of PCD, and similar thing was observed using the transcriptome analysis. As we know that due to the excessive use of antibiotics in various food and agriculture industries, there is a emergence of antibiotic resistance pathogens. So we have isolated a broad host range phage against Salmonella enterica, different serovars, typhimurium, typhi, and enteritides, and this phage has been characterized. And similarly, I have isolated these Aeromonas phages against different pathogenic species of Aeromonas, and their genome was sequenced, and they have been characterized. And overall we found that there was absence of antibiotic resistance, virulence, and temperate lifestyle genes in them, and they are quite stable at different conditions. So these phages can be used for our for making the cocktail which can be used for controlling these pathogens. So in the end, I would like to thank our director, food head of our division, and colleagues from FTD for making so much of work. Thank you.
Thank you very much. 2 quick questions, please.
Yeah, I have a quick question. This de-bittered juice, this nutritional values, have you checked?
Yeah, they have been checked, and they have been notified in that document, technology transfer document.
What is that component which contributes to this bitterness? I mean, what is that?
That I cannot tell.
Okay, that's a
because that is the IP part.
Okay. Okay, fine.
But it has been. It is a chemical which has been added to them. That is.
Okay, thank you.
Any other question?
Is BRC working on vegan meat?
Yes. Recently, we have started in last 1 year.
Okay. So whether any technology has been developed or
No,
it is
no,
in the
no,
process?
no. Right now, there is no technology for the vegan meat.
Okay. Thank you.
So if you had given us the bitter gourd juice and what is the
Meat.
juice
Hmm?
and the banana juice when we visited your facility, no, I would
Yeah,
have vouched for the quality.
yeah.
You did not give us.
Maybe the samples may not be ready at that time.
Thank you. And I thank all the speakers for excellent science. And I'm sorry I had to rush you because of the time constraint. Thank you very much. Let us thank all the four speakers.
I request to Professor Prasanna ma'am to kindly present a memento to Professor Vandan sir. Also, I would like to request to Dr. Majumdar sir to please come onto the stage and present a memento to the session chair, Professor Prasanna. Thank you, sir. Thank you, ma'am. Now let us take a break for tea and we'll get back here at 11:45 for the next session. Okay, there is also an announcement that all the dignitaries and students who are willing to visit the RRCAT laboratories are requested to give their names to the volunteers. And they must carry their institute's ID cards during the visit, and mobile phones, USB drives and other electronic devices are not allowed inside the laboratory area. So keep a note of that. Welcome back. Let us resume with our ninth session of today. This session will comprise of 4 invited talks. It will be my pleasure to invite Professor Abhijit Dey onto the stage to kindly session this chair. And before that, I would like to give him introduction to the audience. So Dr. Abhijit Dey is from ACTREC TMC Navi Mumbai. He is engaged in multidisciplinary research involving preclinical molecular imaging for understanding of breast cancer biology and testing, experimental therapeutics, and cancer nanomedicines. He has been awarded with ICMR International Fellow Senior Scientist Award 2015. He has 110 research publications, review, book chapters, editorials. He has filed for 10 patents, out of which 3 has been granted. He is a member of editorial board, Scientific Report, NPG, Breast Cancer, Dope Press, American J Nuclear Medicine, and Molecular Imaging and Frontiers in Molecular Diagnostics. He has life membership of Indian Society of Cell Biology, Indian Association of Cancer Research, and Indian Society of Nanomedicines. So with this remark, I invite you, sir, and I hand over the mic to you for further proceedings.
Yeah, good morning. We are still in the morning, and this is last but the one session before the last session. Although all of us are expecting that this will continue, so probably the counting of last doesn't matter. So let's the enthusiasm prevails, and we'll go forward on session 9. And as it was announced already, we have 4 talks, and of which we will start with the first talk from Professor Rahul Siddharthan from IMSC. So this is an introduction on the display. His research interest includes transcriptional regulation, chromatin biology, algorithms for NGS analysis, and big data, machine learnings, as well as in the area of health and disease. So with that brief introduction, I invite Dr. Rahul to deliver your talk, please. Thank you. And as per the session designed, the talk time will be 25 minutes, plus 5 minutes of discussion.
Okay. Yeah. I'll use this, it's fine. Oh, change over here. Oh, okay, good. Thanks. So thanks, Professor Abhijit, for the nice introduction, and thank you to the organizers for my first trip to Indore, not just to Hierarchical, but to Indore. It's a very nice city and a beautiful campus, and it's been a fantastic time. Okay, so I'll try not to let myself get told that the time is up. I'll go fairly quickly, and there's going to be a broad overview of a few things that I work on. I'd first like to highlight the collaborators that I've been having over the last 5 years or so. Rakesh is a student who's just submitted his PhD thesis. Ankit submitted a few years ago, and he's a postdoc at Würzburg, previously in Israel. Chandrani is here, and Pavitra is also working with me. So the ones in red are the ones whose work I will mainly talk about today, and the others I will mention briefly. These are various collaborators from other institutes, and they're all fantastic people, fantastic collaborations, extremely rigorous and careful, but at the same time collegial, and I think the students will also agree everybody is an equal in our collaborations. I wish all collaborations were like that. Okay, so without further ado, a few introductory slides on the basics of chromatin and DNA, which you all know. So I'll basically skip past them, but as you know, DNA is basically packaged at multiple levels, starting with these histone octamers, which forms as beads on a string nucleosome model. And these then further packaged into this 30 nanometer chromatin fiber, and there are further layers of packaging until you form chromosomes, which are This is the eukaryotic genome, of course, which gets compacted into the nucleus. And these are how genes are turned on and off via transcription factors, which are DNA binding proteins that recruit or inhibit the RNA polymerase. And transcription factors can bind in a promoter region, or they can bind much further away in regions like enhancers. But they are further away in sequence space, but in 3D space, they come close to each other. And unlike sequence, which is pretty much conserved across all cells in your body, the 3D conformation of chromatin varies serotype to serotype. And there's a lot of other complexity in transcriptional regulation. There are insulators. There's the CTCF, which is an insulator binding factor, but also remodels chromatin and does various other things. Moving on. Yeah, and there are many probes these days to look at all this at the genome-wide scale. This is a slide from the ENCODE project a few years ago, but these are various things that people can look at. How's the genome, how does the genome talk to itself among large length scales? So these high-C techniques do that. They tell you which parts of the genome are in contact with one another. There are various assays that tell you what parts of the chromatin are more accessible or less accessible. There are assays that tell you which proteins might be binding to. So you can chip for a particular protein and find out genome-wide where it is binding. There are assays of methylation and so on. And then the computational prediction part maybe comes in. And of course, there's also RNA-seq and so on to look at gene expression, which we won't talk about. Primarily, we'll be talking about ChIP-seq and Hi-C and how they interact. So first of all, there is this nice video on YouTube that many of you must have seen from the Murney lab. This is a simulation. It's mostly kind of, there's a lot of experimental evidence for it, though you can't see it exactly. The idea is that you have cohesin rings through which the DNA gets pushed, and that's called loop extrusion. And you have oppositely oriented CTCF molecules, which are on two ends, and when it hits those, that's when the loop extrusion stops. And so high-C experiments basically probe how different parts of the genome might be talking to each other. And they do that by basically cross-linking DNA and then doing an intramolecular ligation, and then you reverse cross-link so that these blue and red, which are in different parts of the genome, are actually part of one sequence, and then you do sequencing and a mapping. I'm simplifying it greatly. There are various variants. We'll be talking about Hi-C, which is genome-wide, and in particular a variant called ChIAPET, which in which one of the ends is tagged with a protein. So you're only looking at, for example, it's tagged with Pol2, and you're only looking at what Pol2 is interacting with in the rest of the genome. And this is the output of Hi-C data. You visualize it as these heat maps, which tell you roughly where which parts of the genome are talking to which parts. So the diagonal is the brightest, but you have these triangles of diagonal, which are referred to as topologically associated domains. Okay, so let's get back to ChIP-seq. ChIP-seq is an assay which, as you know, probes where a protein binds, and these transcription factors are known to bind to motifs, short patterns, which are like weakly conserved strings. But when you look at ChIP-seq and you look at a whole bunch of peaks for ChIP-seq and you search for the motif of the factor you're looking at, you often don't find that motif. But what you do find is a bunch of other motifs. This is from a paper by Hunt and Wasserman in Genome Biology a few years ago. They call these extraneous motifs zingers. In particular, there were like 4 or 5 motifs such as CTCF and DTS and a few others, which consistently occur as a fraction of ChIP-seq motifs, no matter what factor you're chipping for. This is kind of their explanation for this, that, well, so they are kind of focused on the cohesin ring and or polycomb or whatever that interacts with this, but So that essentially means supposing you're chipping for the blue factor, because the complex is like this, the piece of DNA that you pull down might actually have a motif for the pink factor, either in trans or in cis. And it might also not have a motif for the blue factor at all, because the blue factor is binding indirectly as part of a complex, and there are all these other complexities. And this is true even if it's not near a cohesin ring. I mean, anywhere a TF binds, it could be binding directly or indirectly. You could be finding no motif for the TF you're looking at, but motifs for the for other TFs. And so if you want to analyze ChIP-seq peaks, there are various pipelines. So, okay, so moving on, this is a paper we published a few years ago. This is basically a method to analyze large ChIP-seq data sets. And so if you have, the problem of motif finding in sequences goes back to the early 90s, and standard tools like NEEM and GIP sampler and so on. And the trouble is they don't scale when you have a data set of 10,000 or 100,000 ChIP-seq peaks. So MEME uses various heuristics, and there are some newer tools that work faster but less accurately maybe. What we thought of doing is think of all these reads as some elements in some abstract space, and we're clustering them essentially. So we're clustering by sequence similarity. It's a hierarchical clustering, and the usual form of hierarchical clustering, which is agglomerative clustering, is too slow because it's of order n squared log n. When n is 30,000 peaks, it's too much. So we do a sort of top-down clustering where we split up a large cluster into multiple pieces. So I'm going rather fast through this because I just want to give an overview. But this is basically the algorithm that you start with everything in one cluster and you break it up into smaller clusters. And and there is a significance criterion. So if you feel that you can't break it up anymore without, I mean, the significance basically, are you doing better than you would with a random cluster? And when you can't, that's when you stop. So this method, though it's not explicitly a motif finder, it actually pulls out motifs very efficiently in synthetic data compared to a bunch of other programs. This is how effective it is. So I should mention that MuMod, the red and green, they love these earlier programs which are more accurate than thickfeed arguably, or roughly as accurate, but quite a bit slower because they are maybe statistically more careful. In terms of speed, this is a very fast program even compared to Chipmunk and so on, which was at that time the standard. On real data, we find some very interesting things. First of all, if you're looking for a particular factor, you do find extraneous motifs like this. So this is an IRF, this is a REST. So this is the, on top in this panel, there are many examples on my website, but these are 4 interesting ones. The top one is the canonical one from Jasper. And you see that there is a secondary motif here, for example. Sometimes there is here also there is a primary motif, but there's also a secondary motif. There is a strange motif here, which you could call it a zinger, but well, it is actually sp2 motif. It's associated with promoters. And there is another motif like this. I don't know whether to call it a motif, but it occurs frequently in many data sets. Another interesting point is that the rest motif is a heterodimer. It's a gap motif. There is a core motif and a gap in another. And we recover another variant of that motif with a spacing, which is known in the literature, but most motif finders can't find it because this is too long. How it was found previously was people look for copies of the first half, and then they look for copies of the second half and put them together. Right. So essentially, looking at ChIP-seq data using this tool, you find a lot more complexity than one might suspect. And the sort of signal for the motif extends much more longer than the core motif, extending sometimes to secondary motifs. We get zingers, so to speak, such as Hunt and Wasserman mentioned. These are factors that they did not mention, IRF1, sp2, GATA1, and so on. They appear in ChIP-seq data sets for multiple different factors. And again, this rest motif appears even in its spaced form in ChIP-seq for sp2. And compared with other programs, we find pretty much the same motifs as the other, but some of these motifs, like the gap one, we are unique. The secondary motifs that we pick up are rather hard to pick up by other programs. Peak motifs, which is quite widely used, actually gives a proliferation of motifs, but later I talked to one of the authors and he said they have a post-processing step to reduce that. Anyway, so that's basically the first part of my talk. And the message here is that there is a lot of complexity even in ChIP-seq data. We are currently kind of revisiting this program and broadening it to other kinds of data, not just sequence data, but tabular data and so on. That's another. Yeah, okay, so is there any biological significance to these motifs? I left this out. So this is ZNF143. It's a zinc finger factor, but most of these motifs are actually the CTCF motif, which you might recognize. It's known to interact with that. This is one of the Zinger motifs that Hunt and Wasserman mentioned. And CTCF occurs in these different variants, one of which has a strong secondary motif, which also has been previously documented. This is like the rest motif that we saw earlier. This is the sp2 motif, and this seems to be some featureless thing. If you look at various biological features, nucleosome occupancy, it looks like these variants of CTCF tend to occur in closed DNA where there is strong nucleosome occupancy. This variant of CTCF tends to occur in relatively open DNA. And if you look at the sort of background, there's a larger GC content, so this is likely associated with promoters, which would lead to the next part of my talk, maybe. And there are other such features you can look at from the level of conservation. All the CTCF motifs are reasonably conserved, but the sp2 one is not. And the distance from nearest TSS, this CTCF motif is closer to TSS than the others. And sp2 motif also is promoter associated. Right, so that's one of the interesting projects we did. Moving on to chromatin, how do transcription factors interact with each other in 3D chromatin? So this is on bioRxiv a couple months ago. It's under review, and it's on the revised version is under review currently. I'm hoping we'll learn something soon. There is a tool that you can try out if you want to, if you have such data. The basic situation is, supposing you have Hi-C data, or in our case we use KIAPET mostly, and you have ChIP-seq data for a bunch of factors, then you can ask the question, do transcription factors tend to occur? So you have one loop of DNA and another loop which is far away in sequence but close by spatially because of the conformation. And you look at heterotypic transcription factor binding. Do they tend to bind, if you see factor A here, are you more likely or less likely to see factor B there? And you can ask the question for all the factors for which you have ChIP-seq. So we have some 30 ChIP-seq factors for the cell line GM12878, and we have both KIAPET and Hi-C data. Basically, we compare how often they actually co-occur in our data with 1,000 randomized networks. And I won't go into the algorithm, but essentially we are preserving the degree of each transcription factor, how many regions it binds to. For example, TF1 binds to 4 regions, the randomized one also binds to 4. And we are also preserving the degree of each of these regions in the KIAPET data. That is, how many regions does it talk to? If it's an isolated region, we map to another isolated region, and so on. So keeping a bunch of these constraints in mind, we generate 1,000 randomized networks and we calculate p values, and then we convert them to q values using Benjamini-Hochberg. So less than 0.5, 0.05 is considered significant for either attraction or repulsion. So in this kind of a heat map, we plot green as significantly attracting and red as significantly repelling, meaning they tend to occur very strongly associate or very strongly don't associate. This is for GM12878 cell line, and this is for K562. The slightly different but overlapping set of factors. The key observation, this was also observed using a different method a little bit before we did it by Ma et al. Essentially, that factors in GM tend to break up into 2 groups. These guys attract each other, these guys attract each other, and they mutually repel the other group. And that was an interesting observation when they made it. We also find that if you look at this square here, this is actually the cohesion factors SMC3, RAD21, and CTCF. And they tend to attract each other as well as interact with some of the other factors there. For K562, it's slightly different picture. You have this group is pretty much the same as this, but the other mutually attracting group is kind of broken up, and that we find in other cell lines, but we don't have so much ChIP-seq data for those cell lines. So we find that this biclustering actually is preserved across multiple. Okay, so this is essentially the same slide as earlier. So we can also ask, do these factors also bind similarly in sequential space? If you ask, do two factors co-occur in a promoter? This could have been asked much earlier. You don't need Hi-C for that, and it's not cell type specific, but well, it is to the extent CTCF is. We find that the pattern is pretty similar. So this is a comparison heat map where the light blue means they completely agree, and dark blue means that one of them is significant and the other is not. Black means they disagree. And mostly they either agree or they only vary in the level of significance. Is there any significance to these two groups? We find that they differ in PPI interactions. These are from the HIPI database, we draw the shortest path of protein-protein interactions linking pairs of factors, and then we ask which are the most likely intermediate nodes within those. And we find that there is a big difference in group 1, group 2, and intergroup. Certain nodes like ZPB tend to, well, this actually shows up in the rest, but these blue nodes tend to show up mainly in group 1 and the pinks mainly in group 2. So the PPI interaction pathways are different. Group 1 is significantly closer to TSS than group 2. Typically on the scale, most of them are within the 1,000 base pairs of the TSS, much more spread out. If you look at predicted target genes, essentially the closest downstream gene to the binding sites, and then look at their possible roles, we find that group 1 is significantly enhanced in housekeeping genes. This is again for GM12878. So I'll kind of skip past this since we are okay. So basically group 2 also is in this case enriched for immune function for GM. But if you look for K562, it's actually different. There was not really a group 2 in K562, but if we treat everything else, which is not in group 1 as group 2, then we get a different outcome. And finally, we find something interesting which had been observed by Sridhar earlier in homotypic factors. So the idea is, supposing, and this has been discussed in individual sequence for many years, if you have a factor binding to a site and you have another site nearby, does it help the second factor to bind? Is there some cooperativity there? And usually there is, and there is kind of a theory for that, that if you have multiple binding sites, then you have more robustness to mutations and so on. So here we are asking an analogous question that if you have spatially separated regions, you have a ChIP-seq peak on one site, it has actually got interacting neighbors via Hi-C or Japet. What is the motif strength on the site that you're looking at if it has neighbors or if it doesn't have? And if it turns out that the average motif strength is, the blue is significantly higher if it is isolated than if it has partners. And that suggests that if you have partners, you don't need such a strong motif, you will bind more easily. If you are isolated, then you are likely to require stronger binding. So these are the conclusions from this part. Thanks. TF-TF associations form into two distinct groups, and these are fairly robust across cell types, across sequential or spatial. And I didn't show it, but even if you just look at motifs, we still see such a pattern. And they are functionally different in terms of proximity to TSS and PPI and all that, as well as in the likely downstream targets. So this suggests there's some complex pattern of spatial cooperativity that has co-evolved with the genome. So in conclusion, I'll just quickly highlight some of the other work that I've been doing, which was the black themes in my collaborators. So one of my major collaborators over more than, I think, 15 years now is Kaustav Sanyal and JNCSR, and these are some recent papers with his group. he mainly looks at various yeast species and their chromosomes and particularly their centromeres. And so this is recent work which is basically led both by his postdoc Ashwati and by my student Pavitra. And they found a way to quickly identify the strain of recently interesting pathogen Candida auris. And these are various larger analyses of centromere function. This is a paper that looks at how replication origins cluster in Candida albicans. And this was actually a nice collaboration, not just with Kaustav, but and Dilaktin Arlikar, whom I mentioned, but also Ranjit Padintiri of IIT Bombay, who did some biophysical simulations of chromosomes. So then I'm interested, so this is work that I should not be showing because this is entirely done by Vishaka Datta, who was a student of NCBS at that time. He's now in NCI. But I acted as a sort of informal advisor. So he first found a way to detect cooperatively bound PFBS just using ChIP-seq peak intensities. So it's a very interesting statistical approach. If you have ChIP-seq data and you have two nearby transcript factor binding sites, are they cooperatively binding or are they independently binding? And as a fallout of this work, he developed a comprehensive simulation pipeline for ChIP-seq, which is useful in many contexts if you're developing algorithms and so on, especially. And finally, I'm interested in machine learning and clinical outcomes, and this is a very recent piece of work. Some of you may have seen Chandrani's poster on fetal growth yesterday. We've also been working with a group in Apollo hospitals on predicting, this is all of Apollo hospitals, and it also includes people from IIT Madras and so on predicting outcomes in the ICU for patients who have recently undergone heart-lung they've been put on heart-lung machines, which is a huge insult to the system. What is the risk of developing systemic inflammation, SIRS? And this is a work with some people in the UK on predicting follow-up to gestational diabetes screening, to postpartum glucose screening for people who have had gestational diabetes, which is an important public health thing. So yeah, that's what I wanted to share. Thank you.
Thank you. That's a very exciting areas in the post-genome era. So it's a very important areas and applications that you are developing. So the talk would be, I suppose there will be a lot of questions, but for the sake of time, I will take 2 questions, pressing questions. Please raise your hand.
Yeah, Rahul, this Candida auris detection here.
Yeah. Okay. Yeah.
This what sort of data base you used for detection? You said it could detect it pretty quickly.
Sorry, I couldn't quite the Candida.
The Candida auris detection.
The clade identification.
How quickly? I mean, you said it quickly. How quickly? And with respect to what?
It can be done using a PCR of certain loci. They've basically identified so basically all these strains are extremely similar, but they've got some significant rearrangements. So first of all, it fell out of centromere work that was previously done on the species. It turns out that centromeres are hotspots for recombination, and a lot of places the break of synteny occurs there. So for PCR, you need primers that are unique to one species, and essentially that's what they found. It's not like very advanced work, but it was useful, I guess. Yeah.
Thank you.
Also, it turns out that these are associated with transposons, and that's something they're looking at now. Many of these, centromeres tend to be associated with inverted repeats and so on, and many of them seem to have transposon origin, or they are annotated as transposons. So that's something that Ashwati and Pavitra are currently looking at.
Any other question? One.
Rahul, it's more of a technical question. So the Twitch feed, what you are talking about, is it a web application that you have, or you have to?
Well, right now, there was a web application, but right now it's not running.
Yeah,
I'm going to put it back up. Okay. The code is available for download, and you can get in touch with me if you want to run it.
Okay. With that, I thank you, Professor Rahul, for excellent talk. And
I request to Professor Abhijit sir, the session chair, to kindly present a memento to Professor Rahul sir. Thank you, sir.
okay. So our next talk is by Professor Abdul Rahman from NICER, Bhubaneswar. His research interest primarily is membrane remodeling, mechanism and regulations of nuclear expansion, structure function, and nuclear dynamic related proteins. His scientific recognition, for the sake of time, I will just point out the most recent one. So he's an executive committee in the Society of Biological Chemistry. He's postdocs in Cornell, and so with that brief introduction, I like to invite Professor Abdul to start your talk. Thank you.
Okay, thank you. Let me thank organizers and HBNI for such an wonderful opportunity to interact with a diverse group of scientists here. We are enjoying this campus, beautiful campus, as a lot of people have said already, and also this hospitality and this wonderful group of people. So we work on membrane remodeling, particularly nuclear expansion and so today I'll be talking about mechanism and regulation of nuclear expansion, particularly role of a dynamine-related protein in this process. So as we all know, nucleus is a double membrane structure. So it has two membranes. The outer membrane is continuous with endoplasmic reticulum, and the inner membrane attaches to nucleoplasm, chromatin, and laminin structure. The nuclear membrane forms the barrier between cytoplasm and nucleoplasm. However, the passage is allowed through the nuclear pore complex. Although there is a nuclear membrane, it's not static. It's always undergoing continuous remodeling. One of the drastic remodeling is metazoan nucleus, when during the cell division, the nucleus envelope completely breaks down and reforms at the end of anaphase to form new nucleus. This is an extreme case of nuclear remodeling. However, there is a stage of this. After division, the nucleus, again, when it enters into the cell cycle, there is a stage called interphase where nuclear content becomes double. And to accommodate this increased volume, the nucleus has to expand. And this mechanism of nuclear expansion is not known in any organism. And this expansion requires incorporation of already with new lipids as well as proteins into already existing membrane. So this membrane is already there. How this new lipid, which is synthesized, gets incorporated into the membrane is not known. We use a single cell organism called Tetrahymena. It's a single cell ciliate. It has two nuclei. One is germline, which is we call mic, it is small, and it is diploid. It has 5 chromosomes. Whereas there is a large nucleus, which we call macronucleus. It's a somatic, and the entire phenotype is governed by this macronucleus. And the ploidy is 45N on an average, although it goes up to 64, and then they divide actually amitotically, so their distribution is not equational. There is the random assortment, and on an average, they maintain 45 copies of each gene. So, but this organism undergoes 2 different kinds of propagation, vegetative division, where the cell in vegetative cell division, they undergo binary division, there is a specific stage called sexual conjugation, where under starved condition in vitro, in the laboratory, two different mating types of single cell tetrahymena comes together, forms a conjugation pair, and there leads to a number of nuclear events, starting from meiosis, where we get 4 haploid micronucleus. Only micronucleus will undergo division. So out of these 4, 3 gets degenerated. One is selected and divided mitotically to give 2 identical haploid micronuclei, gets exchanged between the partners, and mixes genome, they fuse. So here genome mixing happens, 50% from each partner, and these will be identical in 2 parents. Subsequently, this undergoes 2 rounds of mitosis to give 4 identical micronucleus, that is small. And then out of these 4, this is very interesting, although they are identical, 2 are selected and expands profoundly. The volume increases 10 to 15-fold from mic to mac transition. So this mac, which is somatic, will take over the phenotype, and the old mac which was there, it will be degenerated. So always the mac comes from the mic, and that's the way it maintains its integrity, genomic integrity. So the factor which actually is responsible for this transition was not known. We discovered the first factor, which we found to be a dynamine-related protein. So what is dynamine-related protein? It's a group of large GTPases which performs membrane remodeling functions. And there are number of dynamine-related proteins. The classical example is endocytic dynamine, which performs endocytosis from plasma membrane, where it forms a vesicle, which is shown here, and dynamine is essential, which self-assembles on the membrane in this neck, and it's a GTPase. GTP hydrolysis leads to fission of this vesicle from plasma membrane. This is the classical endocytous dynamine, which has 5 domains, which is listed here. There are another group of dynamines, which is called dynamine-related proteins. There are a number of dynamine-related proteins which have different functions. And the main feature of this is it lacks 2 domains, particularly pH domain, plexin homology domain, which is actually responsible for target membrane binding, and PRD domain. The DRP, although they lack pH domain, they also associate with membrane and perform membrane remodeling function. So if you see, there are a number of dynamine-related proteins which performs different functions. As I have already mentioned, endocytic dynamine, which performs membrane fission function. Then there is DRP1, which is mitochondrial fission dynamine, that was chloroplast fission dynamine. And there are another group of dynamine which does exactly opposite function, which is membrane fusion. So mitochondrial fusion is carried out by another group of dynamine. So one dynamine cleaves that mitochondria, and another group fuses. So this dynamics is maintained mainly by dynamine. There is also dynamine which deforms the Golgi vesicles, which is spherical, and it deforms, makes tubular structure and becomes dumbbell separate structure, which is essential for cell plate formation in plaque. There are also some non-canonical dynamines, which actually doesn't perform membrane remodeling function, but it gives antiviral function. But they can also associate with membrane and deform the membrane. If we see, although they look very different from each other, fission, fusion, tubulation, but all the underlying mechanism, they all carry out is membrane deformation. So we can actually classify them as a common membrane deforming agent. The protein DRP6, which we work on, there is something wrong. The protein which we work is a dynamine-related protein, DRP6. No, this one. This one. So tetrahymena, although it's a single cell ciliate, it has a large number of dynamine. Out of these, there are 8 of them, and that corresponds with the membrane complexity in this organism, which is as high as human. We have identified which is associated with endocytosis, 2 mitochondrial, but there is a group of dynamine, DRP3 to 6. They don't have any homologue in other lineages, suggesting they may have evolved a novel function in this lineage. And DRP6, which we have discovered its function in nuclear expansion. So this nicely, when we express GFP tag DRP6, it goes to nuclear envelope. Also some puncta, which we have shown if they are derived vesicles, and when either loss of function by knockout or inhibition of function by dominant negative allele, both inhibits this transition from MIC to MAC, suggesting that this molecule is responsible for MAC expansion. So that was I have discovered during my postdoc. After coming to NICER, we started understanding the mechanism, how it actually brings about nuclear expansion. So as I told, the DRPs do not have pH domain, but they associate with membrane. So first thing, what we addressed is, if this protein has a membrane domain, which we did by expressing and purifying the protein and doing an in vitro lipid binding assay, and it indeed binds to 3 lipids, particularly phosphatidic acid, phosphatidylserine, cardiolipin. And we narrowed down the domain to a DTD, this region, which is equivalent in position with that pH domain. We show by in vitro membrane that it actually requires one of these 3 lipids for association with membrane. It doesn't bind to other lipid. And then we also showed that membrane binding domain is essential for nuclear recruitment. Now, this domain is important for nuclear recruitment. What is the mechanism by which actually this protein goes to specifically to nuclear envelope, not other organelle? So what we have done, there was we actually dynamine, classical dynamine, which actually brings about endocytosis. There was a residue called I533, which is part of a hydrophobic patch, is important for target membrane recruitment. So we did the sequence alignment, but the sequence identity was very less, so we couldn't get any idea. So we did a structural model and found exactly in that position there is an isoleucine called I553. So we thought that which maybe, and that is also part of hydrophobic patch, IMI. So when we muted this isoleucine to methionine, keeping the hydrophobicity more or less same, it actually completely abrogates the nuclear localization, suggesting that this residue actually determines nuclear membrane recruitment specificity. Now the question may be that this is specific to methionine. So maybe other mutation, whether it does the same thing. So what we have done, we have, okay. So before I go that, what we have done, we have used this mutant to understand the mechanism. So if there is a mutant, single mutant, what is defective in this mutant, and that will tell what is required for nuclear recruitment. So since it is a membrane binding protein, we first check the membrane binding ability of this mutant, and it retains membrane binding with phosphatidic acid, phosphatidylserine, but unlike wild type, it has now completely lost interaction with cardiolipin. That's very strongly suggesting that cardiolipin interaction is important for nuclear recruitment. And we have further confirmed in vivo by inhibiting cardiolipin synthesis in the tetrahymena cells. And when we express this protein, it goes very nicely to the nuclear envelope. As soon as we inhibit cardiolipin synthesis, it doesn't go. So that shows that in vivo also, cardiolipin is actually bringing this protein to the nuclear envelope. And it's not because of that nuclear membrane is disrupted because of this chemical treatment. It doesn't have any other defect known for this protein like GTPase activity, oligomerization, and self-assembly into ring and helical spirals. Those are the, this is actually canonical structure for all dynamics. And this mutant can do. That means it's actually only defect is cardiolipin interaction. So if that is the case, then we thought that mutant, if we co-express with wild type, they should be able to self-assemble and can be recruited to nuclear envelope. And that's what we see. When we express, which was not going on its own to nuclear envelope, co-express with DRPC, it goes to nuclear envelope, suggesting that only defect is cardiolipin interaction. Now, to see whether this isoleucine, which we made methionine and loss of nuclear recruitment is specific for methionine or any other mutation will do. So we change it to alanine and it gives a similar phenotype. It completely lost the nuclear recruitment as well as cardiolipin interaction. So one thing for sure is wherever there is cardiolipin interaction, there is nuclear recruitment. Now, the next question, whether isoleucine one amino acid, which is very unlikely, gives the specificity for membrane recruitment specificity. It's a target membrane. One amino acid is there, it gives a target membrane specificity. So this is very unlikely. So to see whether other amino acids also involved, we mutated the surrounding residues and we showed that it's not required for giving the specificity. It can still, mutating can go still to the nuclear envelope and it interacts with cardiolipin. The next question we asked whether the mutant which lost the interaction is due to the change in structure or conformation, but not maybe the direct interaction. So that we addressed by CD spectroscopy and fluorescence spectroscopy. The structure, tertiary and secondary structure looks more or less similar. Still question remains whether there is any local conformation change, which we have actually eliminated by doing quenching study, and it looks very similar. So this set of experiments conclusively showed that DRPC, that protein, a single amino acid in the membrane domain interacts with a single lipid, cardiolipin, to bring them to the nuclear envelope. So cardiolipin has to be in the nuclear envelope. And in eukaryotic nucleus, cardiolipin is not there. But interestingly, tetrahymena and ciliate, other ciliate, they have 3% cardiolipin in their nuclear envelope. And that makes sense why they are adding this cardiolipin. Maybe one of the function is to bring this protein and carry out the nuclear expansion. So if I summarize this part, there is a protein, DRP6, membrane binding domain, one isoleucine gives the specificity to bring it to nuclear envelope. And now what we have done, we have disrupted this interaction by 4 different ways: by inhibiting cardiolipin synthesis or using cardiolipin dye binding to inhibit this interaction, also expressing this domain only to dominantly inhibit this interaction, also only expressing the domain or deleting this domain. All the 4 approaches showed that this interaction is important for this transition. So what we learned is that the DRP6, which is brought to nuclear envelope by cardiolipin interaction, is important for nuclear expansion. So still it doesn't say the mechanism by how it actually brings about the nuclear expansion. So next, what we have done, this is very okay. Yeah. So we presume, since expansion would require more incorporation of membrane, we presume that it may be a fusion dynamic, fission. So we developed an in vitro fusion assay using purified protein. It's only in presence of one protein, our protein, this can give the membrane fusion. That shows that it itself is a fusing agent. It doesn't require any other factor, any other protein. And when we check this liposome, we actually incubate with our protein, only our protein, and check under microscope, transmission electron microscope after negative staining. It can extensively tubulate the liposome, and not only that, it can fuse between liposome. So fusion which we have got is confirmed and appears as a very nice network. It resembles ER structure. So we thought this protein may be involved in ER, maintaining the ER network in the cell. For that, we have actually inhibited this protein function by expressing dominant negative allele and checked that it is actually indeed inhibiting ER network formation around the nucleus. This is interesting. So maybe this DRP6 is bringing the new membrane vesicles, which I have shown earlier, that there is a vesicle, it associates and makes ER network around this nuclear envelope. This is sometimes when we keep longer time, DRP6 shows distribution which resembles microtubules. We thought that it might interact with microtubule, and which we have confirmed by mass spec analysis as well as co-immunoprecipitation, and confirmed that microtubule is also important for its localization to nuclear envelope by treating with microtubule destabilizing agent, as well as using dynein, one motor protein, which actually carries vesicle. Both are required for nuclear expansion and nuclear localization. So this is dependent on microtubule structure and motor protein. So there was a report in vitro They have shown that you can form nuclear envelope in vitro by this extract and incorporating the chromatin. But the expansion requires a vesicular fraction. They don't know what a vesicle, but they need a vesicular fraction along with microtubule and dynein. So this, they propose, based on this in vitro study, that these vesicles, unknown vesicles, are carried by dynein over the microtubule and make a ER network around this, and that is required for nuclear expansion. So what we showed here in this movie, if I show, how do you play the movie? So if you see, this is just GFP tag DRP6, which my protein, and this associates with, this is a dividing cell, and this is closed mitosis, so nuclear envelope remains intact, so this is a dividing nucleus. And it is also cytoplasmic puncta, which is ER-derived vesicles, DRP6 is present in both, and there is fusion between vesicles and as well as vesicles with the nuclear membrane. And this, we think what is happening is this fusion is making new ER network by tubulation and also making new connection with nuclear membrane, giving a membrane source which will give the rapid nuclear expansion. So now the next question, how this function is regulated, because it has to be in a particular state. That we have done by identifying post-translational modification, which one of them is phosphorylation, and one of the phosphorylation is 248 in the GTPase domain. When we mutate to the null, it doesn't go to nuclear envelope, but if we make it constitutive phosphorylated form, it goes more to the nuclear envelope, suggesting that it regulates its localization, and it is by increasing affinity to cardiolipin, which is understandable because cardiolipin is important. We have also shown this phosphorylation increases the fusion function of wild type. This is the wild type and the mutant. So it enhances the fusion function. And this increase in fusion function by inhibiting GTPase activity, because this mutant actually inhibits GTPase activity. And we confirm by giving this GTP non-hydrolyzable to the wild type protein, now it should mimic this, and it actually enhances the fusion function. So it enhances fusion function by inhibiting GTPase activity. We have also shown that this mutant has more tubulating function and makes more ER-like structure, and which could be by stabilizing a particular structure, ring-like structure, wild type, which has more spring-like structure, the transition from ring-like spring structure to ring structure, maybe that is important for tubulation. So with this, the last part I will take 2-3 minutes. So what we have shown, this cardiolipin, which is not present in any other eukaryotic nucleus, but tetraamina with ciliate, it is present in cardiolipin. Cardiolipin is present in nuclear envelope. How it is going? So when we checked the cardiolipin synthesis gene, tetraamina has prokaryotic type cardiolipin synthesis, not like other eukaryotes. And prokaryote cardiolipin synthesis happens in plasma membrane. So we are good that it could be that there is tetraamina, cardiolipin is synthesized in the plasma membrane and carried to nuclear envelope, which we have shown by incorporating culture with labeled cardiolipin, labeled liposome, along with another lipid phosphatidylethanolamin by red. Although phosphatidylethanolamin is present 7 to 8 times more than cardiolipin, when we do the experiment, we see specifically cardiolipin is recruited from plasma membrane to nuclear membrane. That is very, it tells about two novel things. One is there is a novel endocytic pathway from plasma membrane to nuclear membrane to recruit a specific cargo, lipid cargo here, and not any other lipid but cardiolipin, suggesting a very novel pathway. And then we have shown this is dependent on endocytic pathway, which we have shown by endocytic marker, as well as inhibiting the endocytosis, that it is not going to the nuclear envelope. And if I summarize the result, all the result, what we have shown, that there is a membrane domain, which one isolation is important for taking it to nuclear envelope, and this is important for nuclear expansion, which might be by bringing the ER vesicles, which I have shown, bringing them and making a ER network around the nuclear envelope, and gives a nuclear expansion. And also we have discovered a novel endocytic pathway from plasma membrane to nuclear membrane to carry out specific lipid cargo, that cardiolipin in this case. With this, I would like to thank. Oh, I'd like to acknowledge the people who have actually done the work. This work was done mainly by my PhD student Usha and Himani, and last part by Sakti, and collaborators and funding and facilities. And at the end, I would like to thank all of you for your patience. And if time permits, I'd be glad to take any question if you have.
Thank you. We have exceeded time, so maybe one quick question we can.
This is a very nice work. I have just one short question regarding this novel endocytotic pathway that you have discovered. Do you think that some drugs can its pathway and can enter into the nucleus for initiation of the response to drug?
I didn't get that question. You mean that some drug which will inhibit this?
Yeah, maybe there are some drugs which can also goes inside the cell to endocytotic pathway.
Okay, there is some reports now. Actually, recently it's coming like many surface receptor plasma membrane like EGF or GF or something. Those are also actually carried to nucleus. So they are also thinking it could be by endocytic pathway, but there are controversy. They could be direct endosome, or it could be via retrograde transport. So it might end up plasma membrane to Golgi, Golgi to ER, then it goes to nuclear envelope. So there are some evidence that there is a route, but there is not a clear-cut example, and they don't know which actually Pseudomonas endotoxin. That is actually first report. It came in 2018 or something. They have shown an endocytic pathway to nuclear envelope, and they showed by time kinetics that it could be directly from plasma to nuclear envelope.
Okay, thank you. Let us give a big round of applause.
Now I request to Professor Abhijit sir to kindly present a memento to Professor Abdul sir. Thank you, sir.
So, as we start the next speaker, I request we have still two more talks, and I request the next two speakers to please stick to the time, so that the spirit of discussion was pretty high, and it will also remain that way. So our next speaker in this session is Dr. Sandeep Choube, and he's from IMSC, Chennai as well. His research interest includes physical biology, liquid-liquid phase separations, organelle biology. Dr. Choube?
Yeah, so you can hear me. Good. Thank you for the introduction, and also it's been a real pleasure to be here. In general, I think, of course, everyone has said that the campus is amazing, but I find that this auditorium is amazing as well. I was telling Rahul that it's the biggest kind of auditorium that I've ever presented my work to. Okay, so so I have joined. I joined IMSC last year in October, so still early days for my group. So I'll give you a brief overview of what we have been looking at and the kind of problems I tackle. So as you can see from the title, what we are interested in is the kind of governing principles of cellular organizations. Of course, it's a huge topic, and people have been talking about various detailed biological systems and so on. But our approach would be slightly different, as you will be able to see. So, of course, we know that cells are the basic units of life, and they come in different shapes and size and so on. But one thing that all of them have to do is to kind of make precise decisions about which genes to express and at what time under, for different environmental conditions, as well as like organelles such as mitochondria and so on. They have to regulate their number, so homeostasis is another aspect. So these are processes that all of them have to follow. And now what I'm interested in is to kind of trying to find if there are some basic mathematical rules or physical principles that drive these processes. So these are the kind of three things that I'll talk about briefly. And first I'll talk about gene regulation. So on the left-hand side, you see there, what I have is an image. So it's a paper, I forgot to give the reference. So it's a paper from Sanishi's group when he was at Harvard. So they looked at E. coli cells when they're dividing, and they tagged a particular protein. It's lac-I protein, and what you see is that one of the cells is expressing, when it's dividing, one of the cells is expressing that protein while the other is not. So the cells are actually making a decision of whether to express a certain gene or not, right? Now, of course, we know that this is like, I don't have to tell this audience what it means. Of course, through the central dogma of molecular biology, and you produce protein in 2 steps. First, you produce mRNA and then protein. But in the first step of transcription, what you have is a kind of conglomeration or constellation of very different transcription factors, cofactors, and so on. So they come to the promoter and they aid or inhibit the binding of RNA polymerase. Now, these proteins, for how long they are bound and the dynamics at the promoter level is very important for regulating gene expression. So, so let's kind of think of a single molecule, like a single protein. This is the traditional picture. If I'm thinking of E. coli, the picture is that I have a single molecule that can be a repressor or an activator. It comes and binds to your promoter and then regulates your gene expression. Now, if I'm thinking of how much like time it takes for a protein to come and bind to the DNA, or like recognize a sequence on the DNA, like so Rahul had been talking about sequence and all these things. Now, let's say we know the sequence. How long does it take? And then how long does it remain bound to the promoter, right? Now, the simple picture here that I have provided is that there is a simple rate at which it comes and binds, and there is a rate at which it unbinds. So it will give you some sort of an exponential distribution of times, of times for which a protein, let's say, remains bound to the DNA, right? Now, of course, that's a very simple picture, and we know that the nuclear organization is quite complex. Now, so let's just tell you about 2 sets of experiments. One is about a certain protein GRB2, which is part of LAT GRB2 and SOS complex, which is again a signaling, it's associated with a signaling pathway. And of course, when you are making a decision, first you have to decipher what your environment is. And then based on that, you transfer the signal downstream and you regulate a gene. So what happens at the level of at the membrane is very important. Now, what they actually found when they looked at the dwell time distribution of GRB2 is that it actually shows a long-tailed distribution. So what I mean is that if it's a simple exponential, it will just be a straight line like this because it's a semi-log plot. The y-axis is in log and the x-axis is linear. So you should see just a straight line. But it actually shows a long tail here. Now that cannot be explained from your simple picture of binding, unbinding, single molecule binding, unbinding. Now, so that's a caveat and we don't fully understand. And then there is this other paper that last year came out, which was about some proteins related to various processes in cells, including cell differentiation. And what you see is that if you look at the distribution of they call it survival time distribution. I'm not getting into details, but this distribution really looks, long-tailed. And this is something which can be fitted through a power law distribution. Now, power law is, you don't have to understand what it means. Just the main difference between this kind of a power law distribution and an exponential distribution is that exponential distributions die off really quickly, but power law doesn't. So what it means is that if it's power law, you will see many transcription factors which are bound to your DNA for a really long period of time. And these are not rare events. They happen quite often. Now, this is something that can be physiologically very important. If a protein remains bound to a DNA, you can express that like a certain gene of interest for a really long time, or you can keep it switched off for a really long time. So it's really important physiologically that we understand these kind of time scales and the distribution of them. Now, I'm really interested in kind of figuring out what these binding and unbinding times are, how they are regulated. Now, the new thing that has come up, or something it's not quite new, in the 1800s people already kind of had an inkling of this, but it was never really characterized physically. And this is the field of liquid-liquid phase separation. So what I mean by that is that this is a review that I have, a figure from a review that I have taken. And so here you can see that there are all these different proteins, like stress granule, and then you have these signaling punctate, membrane clusters. They all form some sort of a cluster that resemble properties which are like this. If you have a glass of oil and water, they demix, and you see they actually form two phases, right? Now this is turning out to be happening with a lot of bodies in the cell, including various enzymes, proteins, and there's more and more evidence that's suggesting this. In fact, what Rahul talked about when he was saying how proteins bind to a 3D genome, it seems from his data also that there is some sort of indirect evidence of LLP. So it's a very flourishing field, but I'm going to talk about a particular question that we are asking, and that is like how LLPS can let us explain some of these experimental behavior that we are seeing. Now, of course, first you have to see is that GRB2 is part of a cluster which actually looks like this. In fact, if you increase LAT concentration, which is one of the proteins associated with the total complex, they actually form these kind of tiny droplets. It's not spherical because it's more complex. These are multi-component clusters. Again, the physics is quite complicated, but you do see that they phase separate from the system. Now, we also know that from one of our earlier studies that KLF4, which is again a Yamanaka factor, that also forms condensates on the DNA. Now, what do they actually do? What is the physiological role that they play? That's something what you are getting at. Now, again, and this is related to this kind of picture of transcription factor binding to DNA, right? Now, to understand this, what we do, what we kind of see, let's say there is a very simple model of LLPS where a tracer molecule, that's your protein of interest, which is drawn in green, and this is your droplet. Let's say it enters this droplet and you are doing a single molecule tracking experiment. For how long will you see that it's actually stuck in there? Because if it's stuck in there, and if if it's like either on the surface of a membrane or DNA, it's part of the DNA, then you can say that that's the amount of time it's spending close to the DNA. So it can actually activate or repress a gene. So that's why we are interested in this kind of a problem. But of course, there are different kinds of clusters that can form. It can be a homogeneous cluster. You can see actually that there are these concentration gradient of the protein molecules that are forming the cluster. And you can also see that there are these multi-component structures like nucleolus, where you see there are droplets within droplets, and there are multiple kinds of droplets. And so that will make the underlying structure, the inner structure of a droplet very different, and they give rise to very interesting properties in the dwell times. For instance, I'll just show you a few plots. Well, it hardly works properly. Okay, it's not. Okay, so first of all, I should acknowledge that there is a PhD student, Anirudh, and the summer project student, Smriti, who have been working on this project. So these are some of the trajectories that I just wanted to give you a feel for it. So what happens is that a protein enters your droplet, and you want to see for how long does it diffuse. So you write down a diffusion equation. I'm not showing any equations here. You write down a diffusion equation and see how it's diffusing. And you want to see for how long it's stuck in the droplet and after what time they are going out. So that's the time you are counting. And you're looking at distribution of those or histogram of those. So there are some trajectories. Now, if you actually, why is it getting stuck? Okay, now it's okay. Seems like that. Okay. So now the thing is that so I'm just giving you two examples. If I have a homogeneous cluster versus a heterogeneous cluster, you already see that there are some crucial differences in terms of how the distribution looks like. Now, of course, I'm not showing you like it should be characterized better, and we are in the process of doing it. But you can look, for instance, you can look at, is it a bi-exponential or a tri-exponential? You can fit those two, and you can do some sort of Bayesian model comparison and so on. But I just want to show you something more interesting, which is that for now it's okay. So for a concentric sphere, it actually turns out that this kind of power law distribution that I showed from the data, that you cannot get that from this kind of homogeneous, or neither from the homogeneous cluster or heterogeneous cluster. You need some sort of a concentration gradient. So these models we can actually exclude, it seems so far. I mean, still a lot to be done. And it seems that for these proteins that I showed, FOXA1 and others, which showed this kind of power law behavior, it's like this kind of, if you have some sort of gradient towards the center, that can actually give you a power law distribution. And this is something akin to what people see in spin glasses, like, or people understand glass, process properties of glass and so on, where people actually see this kind of power law behavior and so on. So I'm not getting into any of that, but it's very interesting that physically very different systems exhibit very interestingly similar behavior. Now, again, so this is clearly not a complete story, but I just want to, as I said, I just want to give you a flavor. And this is another project where we are now trying to see with a postdoc, Pinaki, and another collaborator of mine, Vani, who is a faculty at IMSC. So we are looking at, if we look at the whole genome, right, and there are these proteins that interact with the genome. Now these proteins also interact with each other. So they can actually bridge various parts of the DNA. Now, how will that impact your organization of the genome? Now, there are, as Rahul mentioned already, there are these high-C data sets where you can see contact maps. And in fact, if you see, we can make some plots like that. Look at the bottom plot. So that's actually a contact map. What we are looking at is a contact probability between different parts of your DNA. When there are these crowders or protein molecules that can phase separate and as well as interact with the DNA. And you can see that we can extract some of this behavior, and the goal is to kind of compare them to high-C data. Still very early days for the project. So essentially, with this kind of LLPS picture, what we're trying to do is to understand how the genome is organized and what the physiological roles they play in terms of controlling various properties of the cell, I mean of the nucleus. Okay, now I'll talk about organelle homeostasis. Again, this is a different kind of problem, but it's our previous speaker already talked about this in much detail. Again, I'll take a very different route. The approach that we have is actually quite simple. Okay. Now we know, before I go on, we know that there are many organelles in cells. I mean, of course, you guys are all familiar with. This is more for a physics or kind of more quantitative crowd. That you have things like nucleus, nucleolus, centrioles, and so on. And of course, cells have to control their number or size. And people have seen beautiful scaling behavior between how the size of the organelle scales with the size of the cell and so on. So those beautiful properties are there, and you need to understand the physical properties of the cell to actually understand the scaling behavior. But we started with a very simple question. And the question had to do with how do cells control? Of course, the broad question is how do cells control abundance, size, position of these organelles in response to environmental cues. I like to think about them as your house. You have a house and you have various furnitures and so on. So you have to really position them well. You would also want to count them. You don't want like 5 refrigerators in your house. So you have to control that. And also the size is important. So all of these are actually problems that cells have to solve simultaneously. You can't solve them can't just solve one problem of number or size or position. You have to solve them simultaneously. And we actually see some evidence, experimental evidence also, where it seems to be the case. But we started with the simplest thing of abundance. So of course you can count various organelles in cells. In fact, these are the three top images are from East, taken from this paper by Mukherjee et al. And as you can see, you can count Golgi, vacuole, and peroxisome. And these are taken from HeLa cells, images from HeLa cells from Mahek Sharma's lab, who is someone I'm collaborating with currently. And so, of course, it's much harder to count cells in HeLa cells. The numbers are really large, and it's a challenging problem to do the image analysis and so on. It's not easy. But in case of East, it's much easier. So you can actually count them, and the numbers go from, like, for instance, peroxisomes numbers go from 1 to some 14, 15 around that. So you really have a good control over that. Now, what do we do with just counting them? Can we learn something about the underlying mechanisms of organelle biogenesis? It turns out that you can, and through this very simple kind of an approach. All you say is that, look, we know that from endoplasmic reticulum, various organelles are produced. Let's say there is a rate at which you produce them, and I call it KD. It's just simple de novo synthesis. And it can degrade. Then they can also go through fission and fusion. We can, of course, also degrade. So this is the simple model we have. And of course, there are different organelles go through different processes. Not all of these processes are associated with all the organelles. But this is kind of a general enough model. But you see that it's a very simple model. You have very few parameters. So you are not really, you don't have to fit 50 different parameters to a model. It's very simple in that way. And it turns out that even in spite of its simplicity, it can actually teach you some interesting biology. So for instance, first let's look at what happens to the organelle numbers. Of course, with these different processes, if it's KD de novo synthesis, you will increase the organelle number. If it's fission, you'll also increase. With fusion and degradation, you will decrease, but the dynamics will be very different, and their corresponding weights will also be different. But using these probabilities, you can actually measure the distribution. You can actually compute the distribution analytically. Now, once you have these distributions, what you do? What you can actually do is that you can look at the distribution really implies in a cellular, in an experimental setup, is that if you have a bunch of cells, you just count the number of organelles of your interest. And then you just make a histogram. Once you have that, you can compute the mean as well as the standard deviation of this distribution. Yeah. And these two are enough for me. So I don't need to know any rates, nothing at all. If you can just tell me what the number and mean and standard deviation is, I can already tell you a lot about the underlying processes. And as you will see, this quantity, which is defined as the standard deviation squared by mean, is going to be super informative. For instance, what you see is that if an organelle is just regulated through de novo synthesis and degradation, it's actually a Poisson distribution. And for which Poisson distribution has this interesting property that the mean and standard deviation are the same. So in other words, it doesn't depend on the actual values of KD or gamma. As long as you tell me the mean, I can tell you exactly what the standard deviation is, right? And so the Fano factor will always be 1. Okay. I just want to show you. So first you see that now that gives me some reference frame. And with respect to that, I can look at other distributions, like other models. If it's just fission-fusion, like mitochondria, it will actually show a noise behavior. Fano factor is a function of the mean, like this. It's very distinct from your de novo synthesis degradation model, right? And similarly, again, it's not. Okay, similarly for other models, you can see various things. Just to give you one example where it can be actually useful, to look at peroxisome, right? And for peroxisome, the traditional model that people think of is fission, de novo synthesis, and degradation. But one important thing is, if that is the case, your Fano factor will always be above 1. It cannot be below 1. But we looked at some data. What we actually found is that it's actually less than 1. So it means that model cannot be true. So our approach tells you that you can exclude model. You cannot predict. It's like this idea of falsification. So you can falsify models, and you can provide really interesting insights even with this very simple model. So it means that you have to look for something else. It's not just those three things. Something else is going on. Maybe there is some feedback or something, but there is something else going on. So that we can say for sure. So that's the power of this approach. So of course, I just want to briefly tell you that, so we had some proposed model, but I just want to tell you, Sandhya Kaushika at TIFR, Mahek at Aizer Mohali, and some other friends that I've been working with, and also Rohit Singh, who is a PhD student. And there is also some stuff that we are doing with gut microbiome data, but I think it's already done. But okay, let's just not talk about it. Thank you very much.
Okay. The talk is now open for discussion. Questions?
Yeah, hello. Yeah, I have one question. Is that you talk about phase separation? Yeah, liquid phase separation. And now when you have a very multi-component transcription factors, now sometimes one factor goes away, another factor comes in into the complex. Now, what do you think of that separation? What triggers of one protein that comes from a different phase of that cell to come into that phase, and what are that in that particular phase that protein goes off? What actually triggers it? What do you think? Because physically, biophysically, it has to be some type of conformation changes or something that will trigger it to come to a different phase.
Okay, that's a very interesting question. So in general, see, the point is that for a multi-component mixture, the physics of multi-component mixtures is still not very well understood. That's the first thing. The second thing is, you have to see there are two pictures. You have this equilibrium picture where you are looking at a droplet in a continuum sense, that it's actually, you're not thinking of individual particles. It's a whole continuum. And you will always have exchange of molecules. Even if let's say I have this mixture of A, B, C that is forming a component, and there is something outside. There is always, something always will happen where you kind of take one single molecule, it can go out. This will always happen. There will be exchange, always. But there are certain properties, like if it's an equilibrium, the chemical potential have to match. So that's a thermodynamic quantity that you cannot, otherwise you are not going to reach equilibrium. So in equilibrium, there are certain aspects of the physics which will have to be obeyed. If you don't obey them, then of course you are not in equilibrium. But your question can also be like what you are saying can also happen due to activity. So if a system is actively driven, it's not in equilibrium or it's reaching towards equilibrium or these kind of scenarios, then many things can happen. And we really don't have a very good understanding of that. But what you are saying is a very important question. In general, traditional phase separation picture cannot naively be applied in biology, because in biology you have many, many components and they interact with different affinity and so on. And also there is ATP and so on to provide energy. So this question that you're asking, it's a very interesting one. I don't know, so I still don't have a very good answer or insight into that.
So in your Fano factor, what is the gamma? Is that also a rate that you are taking into account? And my second part of the question is, would that simplification hold good for other organellar systems, what, like say for example peroxisomes, what you are showing? Would that hold good? Would that be the reason why you are seeing a different trend in your Fano factor than what you expect?
Yeah. So first of all, you'd see. So if I just look at the gamma was the degradation rate. That's all. So if I just take your birth date, so it's called a birth date model. This de novo synth, it's like a very old model, but it has this unique property that the Fano factor remains 1 no matter what the underlying rates are, right? But with the other models, with rates, this curve will change. You will get a different family of curves. But interestingly, it turns out that in the region of Fano factor versus when I'm plotting Fano factor as a function of the mean, there are distinct regions. Like if it's fusion dominated, you cannot be above 1. If it's fission dominated, you cannot be less than 1. So these are general properties that you can get. So that is the aim, basically. That it's like an alternative to other much more complicated and difficult methods of deciphering these kind of processes.
Other questions? Okay. If not, actually, I also have a question, and this talk was actually very wonderful because it's not just talk about modeling molecular functions, but it also talks about the organelle functions. And so my question was related to the transcription factor part, where how does the shelf life plays out in your model? So
Sorry, how does that?
Shelf life. Every molecule has a half life.
Ah, okay, yeah. That's a very good question. So these measurements that I talked about, they go from some 0 to 15, 20 minutes, not more than that. Because in SMT, the problem is you cannot go beyond that. But currently, given the technology that we have. But if you include that, then what it means is that then the molecule that you're tracking is no longer there. So you have to do something about that. I haven't thought about it, but it's a good question. Because if the self life is really tiny, then of course you can't even get the statistics, right? Like the measurements that you're making, because it will just stop after a while. It's just gone. So
see the trackers half-life is actually limiting to understand this 15 minutes.
Yeah, so there is a limit to how much you can, for how long can you track. So beyond that you cannot. So that's the issue. But with these proteins, eukaryotic proteins, typically the half-life is longer. So but the moment you go higher, I think it's becomes an issue. Thank you.
Now I request Professor Abhijit sir to kindly present a memento to Professor Sandeep sir. Thank you, sir.
Okay, so now the last talk is by Professor Pankaj alone. Okay, let me quickly introduce Professor Pankaj. His research interest is primarily in the running out of battery. So I, I will request you to please read the slide rather than I talk to you. And I request Dr. Pankaj to please start your talk. And I will give a warning bell 2 minutes before the final.
Good afternoon, everyone. So, yeah, okay. So, my lab interest is in the translation control, and the key questions what we ask is what is the molecular mechanism of non-AUG start codon selection in the eukaryotic system, and how the non-AUG codon selection regulates gene expression in the eukaryotes. Before starting, I'll just acknowledge the funding given for this work by Department of Atomic Energy, and this work was actually developed with collaboration from scientists from ILS, Amol Suryavanshi, and this is my group. The work I'm going to present here is done by Charles as well as Anup. So the central dogma of molecular biology culminates in synthesis of a protein. And there are fundamental differences between the prokaryotic translation initiation as well as in the eukaryotic translation initiation. So in the prokaryotic system, you see that the selection of a AUG start codon and then the selection of open reading frame is mostly governed by the Chandra-Garneau sequence. But in the eukaryotic system, this is not the case. And the translation initiation machinery scans the ribosome from 5' to 3' direction in the search of AUG start codon, and then it selects the AUG start codon, and from there the open reading frame is kind of a set. So the general pathway is like, in the eukaryotic system, is like the 40th ribosome is then bound by EIF3, EIF1, and EIF1A. These are the eukaryotic initiation factors. And once they bind, you see that the ribosome conformation change from is in the open conformation. Subsequently, ternary complex, which is made up of EIF2 gamma, alpha, beta subunit, binds to the GTP molecule and the tRNA to form the ternary complex. This ternary complex is recruited to the P site of a ribosome in the presence of protein EIF5, which acts as a GTPase activating protein. Separately, you will see that the mRNA is actually bound by EIF4 complex. And then this EIF4 complex is then recruited to the 40th ribosome. Ribosome starts scanning for the AUG start codon. And once the AUG start codons enters into the P site of a ribosome, the GTPase reaction is triggered. And so in this case, like the GTP, the GTP hydrolysis is actually carried by EIF2 gamma subunit, but it is held by EIF5 subunit. And then there is a structural rearrangements happens on the ribosome, where you see that the ribosome goes from the open conformation to the closed conformation, and the tRNA goes from the P out conformation to the P in conformation. And only in this conformation, the AUG start codon is selected, and then the PI is actually released, and then other translation initiation factors are released, and then the ribosome goes into the elongation phase. So we generally use molecular genetic techniques to understand the translation initiation process. And we heavily use these three different genetic techniques, GCN phenotype, GCD phenotype, and the SWE phenotype. So what are these phenotypes? So general amino acid control is related to the expression of a GCN4 protein, which is actually controlled by the presence of these 4 upstream open reading frames. So in a normal condition, what you see that the ribosome binds to this mRNA and starts scanning, and when it comes to this first open reading frame, it translates this first open reading frame, and then the translation is terminated. But the 40th ribosome stays bound, and then it keeps scanning down this mRNA. So subsequently, open reading frame 2, 3, and 4 are actually get translated. And what happens once the open reading code is translated, the ribosome, 40th ribosome and the 60th ribosome dissociates, and then it never reaches the main GCN4 open reading frame. So in a normal condition, you will see that the GCN4 expression is always down. However, in case of a starvation condition, you see that the amino acid levels are down in this case. The tRNAs are not charged, and the uncharged tRNA causes activation of this GCN2 protein kinase, which goes and phosphorylates EIF2 alpha, and phosphorylation of EIF2 alpha causes downregulation of ternary complex formation. And in this scenario, you will see that the ribosome translates the open reading frame 1, and then it starts scanning down this mRNA, but the ternary complex level are rose, so that it doesn't acquire ternary complex till it reaches, it can pass this inhibitory open reading frame 4, and then it can start translation initiation from the GCN4 main open reading frame. And under starvation condition, you will see that the GCN4 expression goes up, and then it can help. So GCN4 is a transcription factor, and it controls around 800 genes, downstream genes, which are involved. Some of them are involved in amino acid biosynthesis and other stress-related conditions. So how does the GCN and GCD phenotype work? So any mutation in the translation initiation factors which does not let the GCN4 expression, even in the starvation condition, is called as general amino acid controlled non-derepressed phenotype, GCN phenotype. And this may be, and this is actually caused by the slow scanning mechanism of or the premature ribosome falloff from this mRNA. The other phenotype GCT is because if you have this GCN2 kinase is deleted and then which cannot phosphorylate EF2 alpha, so in this case you will see that the ternary complex level should be high in this case, and you should not see the GCN4 expression even in the starvation condition. But some mutations in the translation initiation factor causes the ternary complex levels to go down, and the GCN4 expression is constitutively is high even in the normal conditions. So that is what is the GCD phenotype. And the third phenotype we generally employ for understanding the translation initiation is this Sui phenotype, and the Sui phenotype is related to suppressor of initiation codon. Historically, it was observed in this GCN His4 mRNA, in which the first AUG start codon was converted into AUU, and mutations were observed in the translation initiation factors, and it was observed that these mutants were able to translate this mRNA from this UUG start codon instead of this AUG start codon. So, and because of this, the cells can grow in the absence of amino acid histidine supplement. So, we are using these three genetic assays to understand our system. So, our work is mostly focused on this EIF5 translation initiation factors. You see that it has two different distinct domains. Most of these GCD and GCN mutations were observed in this C-terminal domain of this protein, and which were implicated in the scanning as well as the integrity of this complex. But there was only one mutation which is identified at the N-terminal domain, which is this glycine is converted into arginine at 31st position. And this particular mutation was shown to cause a Sui phenotype. That means it can utilize this UUG initiation codon as a translation initiation codon. So that's what you see here, that in a wild type case, the cells can grow in the absence of histidine. But if you have a histidine in the His4 gene where AUG codon is mutated, so the UUG codon is not recognized, and then the cells die. But the Sui mutant can recognize this UUG codon and can grow, although the growth is heavily retarded over here. So it is very slow growing phenotype. And consistently, you see that the UUG by AUG ratio, you see that there is a very high amount of Sui phenotype shown by this EIF5G31R mutation. So this EIF5G31R mutation is very peculiar to this UUG codon, and it does not even recognize a closely related CUG or a GUG as a translation initiation codon. So it is very specific as for the UUG one. So we also observe that this G31R mutation now, and this G31R mutation is predicted to cause a hyper gap activity. But so far, we don't have concrete evidence for that. What we have seen that this G31R mutation, although it's a strong Sui, it also shows a GCN phenotype. That means it cannot grow in absence of histidine, where you are using a 3-amino triazole, which is a histidine amino acid biosynthesis inhibitor. So the cell cannot make enough of GCN4 protein to overcome this. So that's why the cells die. And consistently, you see that the GCN4 expression is more than 50% down in this G31R mutant case. Now, what would be the mechanism for why it is showing a GCN phenotype? So earlier it was shown that maybe the GCN phenotype is caused by the leaky scanning, but this particular experiment actually ruled out that case, in which this upstream open reading frame 1, we have done a mutation and which causes this elongation of this upstream open reading frame 1. So if a ribosome is translating this open reading frame 1, which is out of frame, to the main GCN4, then you should not see much of the expression from this. Okay, but you see that the expression from this is kind of high. So it is not because of the leaky expression. So what would be the other case? We also rule out that it does not have a slow scanning defect. Okay, rather we establish that it has a premature protease ribosome dissociation from this mRNA. Now, why it is actually happening? So we looked very closely to this mRNA and we saw that there are many more UG codons sitting at the 5' regulatory region. So we hypothesized maybe these are the codons which are kind of influencing the GCN4 expression. So in order to test that, we first mutated all these upstream 4 naturally upstream open reading frame, and you see that it does not restore its expression to the wild type level. But once we remove all the other UEG based translation initiation codon, you see that it is kind of a restoring to the wild type level. So it suggests that yes, the UEG codons are actually responsible for this G31R mutation, so that it makes a small short open reading frames, it translates that, and then the ribosomes falls off and it doesn't reaches the main GCN4 open reading frame. So to summarize this, what we have shown that the G31R mutation, which has a strong SUI mutant, and this strong SUI mutation is actually responsible for the GCN phenotype. And because of this, the cell, it prevents the cells from responding to the nutritional stress. Now, since this we have done this in a singular mRNA, now what would be its implication if you see for the whole genome wide? And what we have observed that most, like 45% of mRNAs, total mRNA, has at least one UEG codon at its 5'. So if the G31R mutation is utilizing this UEG codon, then it may affect the whole cellular physiology. Here you see that it can make a short open reading frame from UEG upstream open reading frames, and then the ribosome dissociates and it doesn't reach the main open reading frame of any genes. Or it could create a out of frame open reading frame, and again the ribosome doesn't reach over here. Or it can have a extended N-terminal end. So in order to find this thing out, we did this 2D page analysis. We could find lot of differential protein expression changes, but by doing this multi-top experiment, we could only figure out 4 proteins which were differentially expressed, which were kind of a disappointing to us. Like we thought like there may be many. So we expanded our search more and we used this eye-track technique. And using this eye-track, we could able to figure it out. There are at least 26 proteins which are differentially regulated. 7 were down-regulated, and then 19 were up-regulated. 2 of them I'm highlighting here is Ura6 and then CTT1. And all these genes, which or these proteins which are differentially upregulated, are then we showed in through this relationship map, and you see that they affect autophagy, protein transports, and so on and so forth. And we are kind of focusing on to this. Most of them are affecting cellular stress, DNA damage, as well as this oxidation reduction stress. So to figure it out, like how these these changes are affecting the cellular physiology and what is the mechanism, we checked for this Ura6 mRNA, and we see that Ura6 expression was downregulated in this case. So, and maybe and the Ura6 has at least 4 UEG upstream open reading frame. So what we did, you see that this is a wild type Ura6 expression, and then you see in G31R mutation, the Ura6 expression is way down. But when we remove all these upstream UEG codons, we generally see that the overall expression is going down, but the relative expression in presence of G31R is relatively high. So it is indeed, this is a UEG based expression defect. And Interestingly, what we see that the Ura6 mRNA is highly upregulated in this G31R mutation. So although the mRNA levels are high, the protein levels are low because the translation is probably not happening. And this is what we have established based upon like removing all these up UEG codons, and ultimately we could figure it out that the UEG codon present at this 22nd position is actually responsible for this. Now this Ura6 is gene which encodes uridyl kinase, converts UMP to UDP, and then the UDP is later on used for, which is a precursor for nucleotide, for the DNA and RNA biosynthesis. So if that is the case, then we predicted that it will have effect on, it should show effect on DNA replication space. And consistently we see that most of the genes, proteins which are upregulated is responding to this DNA damage. So what we see here that when we expose it to this 25 millimolar hydroxyurea, the G31R mutation show high sensitivity. But if you overexpress Ura6, you see that the sensitivity is partially restored. And because it has a DNA replication stress, we predicted that it should show a DNA damage, and it was tested based upon this tunnel assay. And you see that in G31R mutation, it shows a high tunnel positivity. Next, we focused on our attention to this CTT1 gene, which encodes catalase T enzyme. And in our case, what we see that the CTT levels are actually high in this G31R mutant case. Now, this tells that maybe the cells are under oxidative stress. And then we check like the expression is high, the mRNA expressions are also high, and when we check the UEG codons present at the 5' UTR region, there are actually 4, 2 in frame and 2 out of frame UEG based open reading frame. And because of this, you see that the expression, although it should be because of the mRNA levels are high, the expression should be high, but it's not going that many fold high, but it's showing a little bit of high expression. And consistently here we see that the cells are like sensitive to H2O2. The G31R mutation is sensitive to H2O2, and if you overexpress CTT, the sensitivity is partially elevated. And we also see that the cells show a high amount of cytosolic ROS as well as the mitochondrial ROS. Next, we have checked that the glutathione levels in these cells are also down, again based upon our ITRACT technique. And one of the other transcription initiation transcription factor YAP1 also show this lot of UEG codons, and again the expression of this YAP1 is also down in the G31R mutant case, which is actually should be responding to this ROS related toxicity. So the ROS related toxicity cannot be alleviated because the YAP1, which is the master control here, is also down, and that's why you see that the cells are growing so slow because of this kind of a stress. And finally, we established that this G31R mutation does not have any defect with respect to translation initiation, but has a defect with respect to the translation elongation. And this translation elongation defect is, AI5 does not play any role in elongation. So why the elongation is down? And previous work from other labs has shown that this profile you can see if there is a oxidative stress, and which is kind of a consistent with what oxidative stress we have also seen. So to summarize our work, what we have seen that the G31R mutation has a pleiotropic effect on the proteome expression, which affects multiple cellular pathways, reprograms gene expression, and it creates ROS toxicity and also DNA damage. So finally, what is the significance of this particular work? Mutation in EIF2B, which is a guanine nucleotide exchange factor, has shown to cause this vanishing white matter disease, which is a childhood ataxia. And this is caused because of the lower level of ternary complex. Another mutation in EIF2 gamma subunit shows a hemo syndrome, which is mental retardation and so on and so forth. And in both the cases, you see there is a downregulation of a ternary complex. But the symptoms are totally different in each case. So when this EIF2 gamma mutant, the mutant which caused the hemo syndrome, was actually tested in yeast, it shows a similar ternary complex defect as well as it shows a Sui phenotype. That means it is kind of a utilizing UG as a initiation codon. So most probably this hemo syndrome disease may be because of additional UG codon recognition, and our study can give kind of a impetus to study further what is actually happening in this kind of a cell. So with this, I thank you all for listening to my talk.
Yeah, excellent talk in the core area of molecular biology, and with a wide application naturally because it's translational heat. So of course we are waiting for lunch, but we will still encourage a couple of questions from the audience. Yeah.
Everyone is hungry.
Yeah, it feels like. Okay. If there is no other discussion to continue, then I thank and please give a big round of applause to all the presenters.
I request Professor Abhijit sir to kindly present a memento to Professor Pankaj sir. Thank you, sir. Now I request to Professor H. Pal sir to kindly come onto the stage and present a memento to Professor Abhijit sir, the session chair. Thank you, sir. Now let us take a break for lunch, and we all will assemble back here at around 2:30.